Hi everyone. We have covered the first main chapter of general microbiology, that is to do with all the historical aspect as well as the stains, which is one of the most important basic things. And thereafter, now we go on to another basic which is very important again, that is microscopes and bacterial anatomy. This is the plan that we have for this class, and of course then we'll be going on further in the next class. As I had told all of you, firstly, the timing shall be 5 p.m. as requested by all of you, because that is when your university classes are over. Secondly, going forward in the month of January, we are now going to increase the number of classes. Slowly, we are making you used to this entire thing. This time in November, December, we gave you one class per subject, but now going forward in January, we will, for example, have 3 classes of pathology, 3 classes of microbiology and of pharmacology. So with me, you will be having total of 6 classes of Path Micro combined in Jan, and then in February, we will again make it a little more. Now we are making this one by one, we are leveling it up in phases so that it becomes easier for you to adapt to this entire thing. So I hope all of you studied. Like I remember last class when we met for the second pathology class, you guys had studied the first pathology section. So I'm assuming that if Path you studied, micro you must have, because what was covered was pretty simple, na? Scientists and stains. I don't think it was a very complex thing that we covered. It was pretty simple, I believe. And I hope you believe so too. And today I'll start off. I've divided my lecture. We are going, other things keep coming up in your entire journey, from making a file to giving exams. You will feel that you are running short of time, and that is when this time management skill of what I call as time boxing will help you. And this is not a skill that I can teach you in one night. This is something I will gradually teach you over the classes. Like for example, today I know I have to cover 2 topics, microscopes and bacterial anatomy. When I open the book, it is huge. When I open the QBank, it is further bigger. So I mean, how should I cover it in just 2 hours? So what I'll say, I'll give you a 40-minute timestamp in which I believe we want to finish off the microscopes. And the remaining time that we have, we are going to finish off the bacterial anatomy. Let's see if we can accomplish this. And at the max, if we don't, we'll be left with 10 minutes lag. We'll extend till 7:10, big deal. So now let's get going. And this, you will actually realize that now when I have in mind that I want to finish the microscopes in 40 minutes, I think I will be able to finish it in 40 minutes with a plus minus 5 minutes. So let's get going. I'm starting with microscopes. I'm sure all of you have seen this microscope in your labs. Have you guys visited your lab? Path lab, micro lab. Both the labs are going to have a microscope. This is famously called as a light microscope. We will come down to the parts, but what I first want to ask you, what all does a microscope have? Number one, it is going to have magnification. Obviously, magnification is because there are so many lenses that are there. Forget microscope. You guys have seen, all of you, any of you who wear spectacles, if you wear a lens will magnify something for you. So I want to magnify. Like for example, you say that you want to look at this cell. That's the only thing I have around me right now. But if you want to look at this cell, you can look at this with your eyes also. But if you want to look inside the cell, you want to see that, oh, this cell was kept over here on the table since 4 months. You feel that a lot of bacteria must have gotten deposited on this cell because it was kept on this table for 4 months open. So now what will you do? Can you look at the bacteria with your eyes? You can't. So you will put a lens and you will magnify this image. And then you will be able to see that if there is a bacteria or a fungus sitting on the surface of this cell. So the first most important thing required is a lens system that will help in magnification. Second property of a microscope, and this is any microscope, you will, what kind of question, theory questions you get, that I will tell you gradually. Pehle bas samajh lo, then I will divide it into sections. You will get to know, okay, this is where I get a short note. You don't get differences over here. You don't get long questions over here. You only get short notes. And you only get practical questions. So, usi point of view se you have to study. Let's start. Magnification done. Second, resolution. What is the meaning of resolution? Anyone? This is not the resolution that you are going to take in another 6 days on New Year's and never going to fulfill. And usually as a medical student, our resolution is always, New Year's resolution is that I am going to be very regular with my studies throughout the year. I will not leave my syllabus for the last day or the last month of the exam. I will study every day 2 hours. We have all kept those resolutions and we have all failed miserably throughout our lives in completing those resolutions, but not that resolution that the microscope takes. Resolution means, before I write the definition, ability to tell 2 points as different. So for example, if like we don't have a microscope in our eyes. In our eyes, ma'am, we have normal lens, normal eye system. As per you, this is one big dot. Do you all think this is one big dot? With your eyes, this is what you can see. But then I decided that let me look at this with a microscope. Let me actually zoom in, and like a microscope, let's assume I'm becoming a microscope in this iPad and I am zooming in. I realized these were not two dots. These were not one dot. These were actually two dots. But when I was looking at it with my naked eye, zoom out vision, I thought it was one dot. When I actually zoomed in, I realized no, there are details, there are two dots. So that ability to tell that two things are separate. For example, the last two names that I can read are Om and Tushar. Okay, so Dr. Om and Tushar, if they are standing in one straight line, I will just feel that only one of them is standing. But if they are standing one behind the other, I will feel only one person is standing. But if they stand side by side, I will be able to tell who is Om and who is Tushar, right? Same way, that is what is called as resolution. It is the ability to differentiate two points as separate. So could your eyes make the difference between the 2 points? No, this means your eyes don't have the resolution, a good resolution. But when you looked at it with zooming in with a microscope, then could you see the 2 points? You could. So what has a better resolution? What could see the 2 points separately? Tell me, do your eyes have a better resolution, or does the microscope have a better resolution? Obviously, ma'am, microscope has lenses. It will have a better resolution. So if I have to compare my eye versus the light microscope that I showed you that you have in your lab versus a third microscope which is even more advanced, I will show you, that is electron microscope. So resolution is said to be, for eye, it is 0.2 mm. 0.2 mm means that your eye has that much of resolution. Any two things that are 0.2 mm away, that your eye will be able to tell that yeah, these are two separate points. 0.2 mm distance is minimum required. Next, for a light microscope, it becomes 0.2 microns. That is how closely it can visualize things. And for electron microscopes, it is in nanometers, even smaller, means that I can vary two very, very close things, which even a light microscope might feel that no, these are not two things, this is one thing only, one dot. But electron microscope will zoom in further and say, no, this is not one dot, these are two dots. So you are seeing that the resolution is going higher and higher and higher. Repeating, I've learnt it best in the form of 0.2, 0.2, 0.2. Only unit is changing. For the eyes, it is going to be 0.2 mm. For light microscope, it is 0.2 microns. And for electron microscope, it is between 0.2 to 0.5 microns. Resolution samajh aa gaya, magnification you've understood. But there is no microscope which can work without contrast. What is contrast? Now, for example, how should I explain? What is the background of the PPT color that all of you are seeing right now? It is black, right? Now, over here, why are you able to, if I, for example, I write with a white pen, all of you can see, ma'am, you have created contrast. The background is black and you are writing with white. So this is going to be a contrast. That is why we could see that star that you have drawn. On the other hand, if I now, you won't even be able to see it, but if I draw it with a very dark color, say with dark blue, you would be straining your eyes a little to see that, yeah, ma'am, I can see one star you have drawn, but it is so dark in color that I can't see it properly. That is what we do in microscopes also. If my microscope, for example, if I'm using a light bulb, if I've kept everything white, do you think I'll be able to see a white color bacteria on top of it? No. Ma'am, if you've kept the background white, then add some pink, blue, nice color to the bacteria so that I can see it. So how do we do that? We do that with the help of dyes. So same way, what we've done, if the background is black, the technical team has made sure that I am visible to you. So they have made sure that this camera section of mine is well lit up. On the other hand, don't worry if my video goes away. On the other hand, if they would have changed the settings to this, now suddenly you would have said that, ma'am, you are not visible. That is because we have created a contrast so that you can see me against this black background. That is what we do in a microscope. So now let's get going. You've understood three properties of a microscope. Jaldi tell me the three names. Number one, magnification. You do that with lenses. Second is resolution power, which means seeing two things as separate things. And third is contrast. Very important. Color theory you will study now. How against white you should use black, black you should use white, opposite colors you should use. Now let's start with the first microscope, that is light microscope. Now tell me guys, what is the source of light? What do you think is the, you'll say common sense ma'am, light microscope mein you must be using a light, a tiny little 2 watt 3 watt bulb you must be using. Exactly, what I've put over here is going to be that light source. This is the tiny little bulb that I add over here. So now if it's actually available in the market, it's that simple a light that you have, the bulbs that you put in your house, a little smaller than that because it's chota. So that's it. Otherwise everything about the light is the same. What will light do? Light can go in every direction. Light will go left, right, light will go in the entire room. Do you want the light to go to the entire room, or do you want that, ma'am, I have, how many of you gone to the path lab and the micro lab? Have you been given those glass slides? Are you placing those glass, have you seen the glass slide, and on top of the glass slide you have some specimen over here? Maybe it is appendix if you are in the pathology lab, maybe it is a bacteria if you are in a micro lab. All of you have seen the glass slide, and the rule in the exam is do not break the glass slide. You break the glass slide, you are definitely going to get penalty over it, and they will not just charge you for it in many colleges, but they might not give you marks also in your final exam. Don't break glass slides. So coming back, okay, yeah, so Shreya says yes, Om also says yes. So you all have seen the glass slide. Todna nahi, my first most advice as a senior to you. So now where do you want? Should the light go in every possible direction? Do you want the light to go everywhere, scattered? Or you want the light should only and only go to where I want to see the appendix or the bacteria? Ma'am, light should go only to the object. So for that, you will have to make sure something is there. First, you should know that this is a slide. And what slide? All of you told me this is a glass slide. Where has it been kept? It has been kept on this thing which is known as the stage. Of course, because the hero always has to be kept on the stage. That is, your hero is your glass slide. Handle with care, glass slide. Hero has to be kept on the stage. Now you will tell me that, and I say indirectly, that spotlight kahan jana chahiye? Ma'am, spotlight has to go to the hero. Spotlight has to go to the stage. So I don't want light should go everywhere like this. I want light should travel only to the microscope, the slide. How should I do that? I have a tiny little round structure below the stage, and this tiny little round structure is known as a condenser. What do you think a condenser will do? Look at the name, condenser. Ma'am, it will condense the light. If the light was going like this everywhere, it will condense the light. It will make it into a tunnel. It will say, okay, no, light will only come this much area. Light will not go here and there. It will get condensed. Who does that in your eyes? Like right now I'm sitting, I have a light in front of me. Light is going everywhere. Light is going in my eyes also. But who is taking the light and putting it on the lens? It is the iris. So can I say inside the condenser, there is also, it's actually a microscope is working on the human eye principle only. In our eyes, we have the iris. Iris makes sure that the light goes to the right direction. Over here inside the condenser also there is an iris. Now the light will go only to the hero. Light goes only over here. After that, where are you sitting? Ma'am, I am sitting over here. I'll make your eyes over here, okay? This is where you are sitting, okay guys? And you are peeping in. By the way, how have you trained yourself? Are you that category of student who is very professional and who is looking into the microscope with both the eyes and can focus? Or are you that student who's closing one eye and then looking at the microscope with one eye? Mostly students fall in the second category, where they can't maintain the vision, because usually our microscopes are designed for both the eyes to view at the same time. What we are trying to do is a binocular microscope or binocular vision, because we used to get one eye. In old days, like 40-50 years ago, people used to actually have only one lens. They used to peep into that one lens with one eye. Then they started having vision problems because only one eye was being used. Then they discovered these two eye lenses so that both the eyes could view at the same time, just like you see the binoculars and you try to see something with binoculars. So can I say this, where my eye is, what is this set of lenses known as? This is known as the eyepiece lens. This is known as the eyepiece lens. So this has a magnification of 10x. Like your phone cameras have the magnification, this lens will magnify 10 times. okay? But I have one more lens system over here. Can we all see how many lenses do you see over here? Single or multiple? I can see, if I count, I can see 1, 2, I can see a tiny little at the back, 3, 4 lenses to here, ma'am. And these lenses are very close to the hero, the object, the slide. So can I say the ones which are close to the object, those are referred to as the objective lens. Are we okay with this? There are two lens systems. The ones that are close to the eye, they are known as eyepiece lens. The ones that are close to the object are known as the objective lens. But ma'am, you said there are many objective lenses. So can you tell me how many objective lenses are there? Total 4. Before that, eyepiece lens, tell me the magnification, guys. Eyepiece lens always 10x zoom karega. Simple. Objective lens is going to have total of 4 lenses. Number 1, scanner. Scanner lens is, ma'am, a very small lens. It can only zoom in 4 times, 4x. Then a little bigger lens called low power, that can zoom in for 10x, 10 times. Then further high power, that can zoom in for 40 times, 40x. And then the biggest lens that you have, oil immersion lens, that can zoom in 100 times, that is 100x. So repeating, 4, 10, milado, 40 and 100. 4, 10, 40, 100. So 4x is just like scanner. 10x is okay, going up, low power, then 40x high power, and finally 100x oil immersion. How many of you have gone to the lab and have you been told, maybe in the initial days you won't, but sometimes you just look at a slide. Sometimes they also give you a small bottle, a dropper with the oil. Have you ever wondered why next to the microscope have they kept a bottle of oil? Because if I start putting, like you take that dropper and if you put 1 drop of oil over here, like I'm marking, just 1 drop of oil if you put on your slide, then which lens will you use for viewing? Ma'am, if I've added oil, then I will be using this microscope lens, the lens which has the maximum magnification, that is 100x. All this that I'm teaching you today, when you will go to the lab next time, please, whatever I've taught you, line by line, go through the points and you will see everything in a microscope. So coming back, now you will tell me that, ma'am, light is passing, this is the light source, then the condenser made sure that the light went on the stage, then the light must have gone to objective lenses, and then the light must have come to the eyepiece lenses, and then I am sitting over here and viewing it. So what is the final zoom in magnification that you are going to see? What is the total magnification that you notice? You just multiply the two powers. What is the power of objective lens multiplied by eyepiece lens? That is your total. So ma'am, for eyepiece lens, the power is always and always 10x. You've told us. But for objective lens, that depends. It could be a scanner which is 4x. It could be a low power which is 10x. It could be a high power which is going to be 40x. And it could be oil immersion which is going to be 100x. So total magnification will depend. If you are using scanner, tell me guys, what is total? It is going to be 4x into 10x. So total magnification will become 40 times. If I am doing 10x into 10x, then it will become 100 times. If I am using 40x, so 40 into 10 will become 400 times. And if I am using 100 into 10, that will become 1000 times. So I realized eyepiece will always remain 10 because eyepiece is only one lens, always 10. The magnification will depend on which objective lens that I've used. So first question they will ask you, how is the total magnification calculated? Just multiply the two lenses. Next question is, what is the maximum possible magnification of a light microscope? Maximum possible magnification of a light microscope is 1000x. You've understood how 1000x came? Because the maximum of objective is 100x and eyepiece is always 10. So the total becomes 1000. This is a very famous PYQ that they ask you. Now coming all the way back, anything left? Yes. You might say that, ma'am, these lenses, they will work differently when you look into the microscope and they will work differently when I look into the microscope, because ma'am, you have a different vision power, I have a different vision power. Maybe some of you have spectacles, you will also have specs, you further have a lens on your eyes, so you will see it at a different level than I will see it. So maybe sometimes you might want to move this slide up and down. You might want to move this stage up and down. For that, you've got two knobs over here. If you see, there is a big knob and a small knob. With that, we can move it up and down. So that if your vision and my vision is different, we can adjust and see the slide. So the big adjustment is known as coarse adjustment. If you feel that you want to move the stage like 2-3 centimeters up, 2-3 centimeters down, massive movements you want to do, then use the big knob. If you say only few thoda blur lag raha tha ma'am, I just wanted to clear it a little, then you are going to do fine adjustment. In fact, any of you who are very fond of photography also works in the similar manner. First, you do a proper zoom in so that you can get the picture and you can get the subject in place. After that, all that you do is, like in your cameras also, you touch on the screen so that wo autofocus ho jaye. That fine adjustment you do so that the blurriness goes. That is how you have two knobs to move the stage. That is an entire light microscope. Now I'll ask you a previous year question. Let's do an MCQ, two MCQs. Number one, what is the maximum magnification of a light microscope? It is 1000x. Ye bata diya. Second question is, stage is present or condenser is present where in a microscope? Is the condenser present above the stage? Is it present on the stage, or is it present below the stage? Tell me, where is condenser present? Condenser is present below the stage so that it can condense the light. This is the second PYQ. I think all of you will be able to answer. The third PYQ, what kind of a light is going to be present? Are you using what kind of light? So you'll be like, ma'am, aapne kaha tha regular bulb jaisa chota sa bulb, white light, that is what is being used. That is known as transmitted light. So now even if you're sitting in a room, do you have any tube light under which you are sitting in a room? A tube light or a bulb, some light source you have? Or if you're looking at your iPad or your computer screen or your mobile, light is coming to you. Ye sab kaun sa light hota hai? It is transmitting in the air. All these are sources of transmitted light. So the light microscope works on transmitted light. Reason I'm telling you this is because now I would want you to understand what is a dark field microscope. Dark field microscope, you will say, ma'am, acha, usme everything was light and bright. You were saying light is there and all of that. Now you will say everything in the microscope will become black. Like this PPT screen, everything in the background will be black because now the light that is you are using is reflected light. Do you get any bulb where they will market it and sell it that, oh, this is a bulb from which reflected light comes out? No. Every bulb that you go to buy in the market will be the same. It will be transmitted light. Means I have got a light source over here and I have got a light source over here. Both of them will transmit light. I will have to convert the transmitted light into reflected light. Let's understand how we are doing. First, I will teach you what I just now taught you in a light microscope. In a light microscope, I taught you this was the light. The light is going to go everywhere. Then that condenser will say, no, the light must go only to the hero, the spotlight. So the light will be sent on to the hero, that is the specimen. And then you will see a white color ka bright specimen, whatever appendix or fallopian tube or whatever you want to see, you will be able to see. Ye tha normal microscope. Can you see what is the difference in a dark field microscope? Over here, what I have done, this is the same light, same chota wala bulb, nothing different. This is the same condenser, but in between the two, I have kept something. I have kept a central aperture. Do you notice what has happened because of that? There is a cone area. Can everyone, before I proceed, can everyone see there is a cone of darkness that I have created over here? I have so nicely manipulated my light. I said that over here, I want background pura dark. Like this PPT slide, the background should be totally dark so that I am the specimen, like I am the appendix today for all of you, or I am the bacteria. I should only shine. Everything in the background should be dark. So what I said, don't send unnecessary light everywhere to the lenses. Don't send any light to the lens. Only send light to the object. Only send light to that one. See, this is a bacteria. You said sirf bacteria ko light jayega. Only the bacteria will receive the light. Then the light from the bacteria will get reflected and then it will go to the lens. That is what is the concept of reflected light. I will teach you once again before I show you the text. You have used a regular bulb, but the difference is you have made a central aperture, that is a cone over here, making sure that your light, does your light go everywhere or does your light only touch the specimen? Does your light go anywhere to the lens? No. Your light only goes to the specimen, your bacteria, virus, whatever, and that will now reflect the light. So as a result, what happens? Now read. What have you used over here? This is known as a dark field condenser. You have used a special type of a condenser called dark field condenser. Now read, because the same thing you will have to write in your notes, in your short note. This is there in your textbook. I've taken it from your textbook. The dark field condenser illuminates what? It illuminates only the object with what? With a cone of light. Could you all see? It created a cone of light and it only illuminated the object. Samajh aaya? Without any light going onto the objective lens. Did it allow any light to go from here to the lens? It did not. Ma'am, it created a cone of light and the light has only gone to the specimen. As a result of which, what I see, background to color hi nahi aaya. Background is total black. Can you see? Only the bacteria is shining, because spotlight, na, this is exactly like spotlight. You have put the light only on the bacteria. Only the bacteria will shine. Rest, background, support staff, all the background dancers, everyone has been made black, mute, gayab kar diya. When do you do this? Number one, dark field microscope kaun se light pe hota hai? It works on reflected light. I do this when I have to see very thin structures. See, if for example, make this dot, you will say, ma'am, this dot is anyway so big. You throw light on it or you don't throw light on it, I will anyway see it. It's so big. But if I have to make you identify something like this, you will say, this is, I don't know if you can see also, but this yellow color dot is so tiny that you will really have to highlight it first, then only I will be able to see it. And that is what you are doing. This bacteria which are very, very thin, and giving you an example, the name of a bacteria is spirochetes. They are so patla, they are so thin that if you make everything nice and bright, you will never be able to see them. You will have to make sure, like the stage pe wo play hota hai, spotlight light jata hai on the hero, on the main character. The spotlight has to go on this bacteria, only then you will be able to see it. Can you see? Spirochete tells me it is supposed to be spiral, and I can see that there is one more structure which is very thin in a bacteria, which I will teach you in a while, and that is flagella. Do you know what flagella is for? Flagella is like a tail. Many bacteria, they have a tail through which they swim or they move. When the bacteria wants to move from here to there, they don't have feet, they have flagella. So flagella are also very patla. You will see them with dark field microscope. Ho gaya? Can I move on to the next microscope? The third one is going to be the fluorescent microscope. Now fluorescent microscope, what is the light that you will use? That is going to be the ultraviolet light. Before that, Dr. Om is asking, how does the light only reach the bacteria? That is exactly what we were doing, Dr. Om. What did I say? You have used a special condenser. In this, did I say that I have created a special kind of a condenser? Ye normal condenser nahi hai. This is a special condenser in which I created a central area of darkness. This is a special device that you get from the manufacturer, from the market, where the light will only and only go like a cone to the object. It will not go anywhere else. So that central darkness, that central aperture is a special extra device that you have added. Then only the dark field microscope will form. Okay, coming to the third one, fluorescent microscope. Now what do you have? You are going, now ma'am, you can't buy any light from the market and put it. In this fluorescent microscope, you will have to use a special light, that is ultraviolet light. If you are attaching a bulb over here, make sure it is an ultraviolet light bulb, number one. So let's start reading. Let me zoom into this now, and I have got simpler. See, these are the photos you can't draw in your theory exams. They are good photos, but you can't draw them. What you will draw is actually what I'm showing you as line diagrams. Only these are possible for us to draw. So what we've done, we have taken an ultraviolet light, and ultraviolet light started going. Where am I sitting, ma'am? Where is the slide? This is the specimen, or you can call it a slide. Chalo, let's assume I am dealing with apni country mein most important infection, TB. Patient has come to me with balgam wali khansi, cough with sputum, and then I want to see, does that patient have TB in his sputum? So this is where I've kept the slide. This is the detector, means this is where you are sitting. These are your eyes. You are looking into the microscope from here. Okay, this is where you are looking at the microscope. Now ultraviolet light comes from here. You will say, first ma'am, you will have to send the light to the slide, whatever you have prepared from the patient. Then the light will go back and it will come to my eyes. Then only I'll be able to see it. First send the ultraviolet light to the slide. So this ultraviolet light, so how can I just, light is not like my friend ke kahin bhi usko left ghuma diya, right ghuma diya. Light will move only when you have certain directions to give. So what I've placed over here is a special type of a mirror so that I can reflect the light, and this mirror is known as the dichroic mirror. This dichroic mirror is very smart. Mirror is, you will say, ma'am, a mirror should, two types of mirror like we used to study in physics. Either it will reflect all the light or it will let all the light pass through. What are the options here? I say when ultraviolet light came, it reflected. It didn't let it pass through and through like this. When ultraviolet light came, it reflected it in this direction. It reflected it onto the slide. Because this ultraviolet light which I have used is of a short wavelength. You all remember physics chemistry mein ye wavelength ka symbol tha. It has used a short wavelength. We are using, the bulb that we take is a short wavelength bulb, and that light will first go to the slide. Let's assume your patient had cough with sputum. Clinically, are you guys suspecting in our country someone comes to you with cough and sputum? Are you suspecting number one thing in that patient has TB? So when you know that most likely patient has TB only, you will add some dye over this. Onto the slide specimen, you will add some dye, and not a regular dye. You will add a fluorescent dye, something that will shine. Fluorescent is usually green, na? So you will add a dye which will shine a little. As soon as the light hits this dye, light gets very excited, and then the light transforms into another light वापस जाने के लिए, and that is going to have a longer wavelength. So now did you see what dichroic mirror did? When the shorter wavelength light had come, dichroic mirror said side ho jao, it reflected it. But when it got converted into a longer wavelength light, dichroic mirror has allowed the longer wavelength light to pass with. You can say shorter wavelength is like general public. Dichroic mirror has put it on one side, but when it got converted into longer wavelength, it has become VIP. Now dichroic mirror will allow it to pass, and who is sitting over here? You. Now it will reach your eyes and you will see something like this. You will see a green color ka fluorescent image. Has everyone understood the principle of a fluorescent microscope? In this much detail you will not find it written in your textbooks, but in a short note you will have to write it, and then you will be unnecessarily looking on Google for it, which I don't want you to. So can I say number one, what is question number one? What light do we use? Ultraviolet light. What is the mirror that is used? It is going to be the dichroic mirror. And what is the principle? The principle is that a shorter wavelength is going to be converted into a longer wavelength of ultraviolet light. यहाँ तक clear हुआ? Listen to this entire thing like a short note from me. We are using an ultraviolet light as the light source. It is emitting a short wavelength light that is reflected by the dichroic mirror to the slide. On the slide we have added a dye which is a fluorescent dye. That dye will convert the shorter wavelength into a longer wavelength which will pass through the dichroic mirror and come to the eyes. Yahan aa gaya longer wavelength. Now you can see. So you will say, ma'am, because in the patient you were suspecting TB, you added a fluorescent dye. What if patient had malaria? What if patient had fungus? This means that for every organism you have a different fluorescent dye. Adding dyes is known as fluorochroming. When dyes are added, please remember, adding fluorescent dye is known as, fluoro means fluorescent, chroma means color. So when you're adding fluorescent colors, that is known as fluorochroming. So if you were suspecting Mycobacterium TB, what are the names of the fluorescent dyes that you can add? It is going to be auramine and rhodamine. It is going to be auramine and rhodamine. These are fluorescent dyes for TB. In the next class of pathology, which is coming up in the first week of Jan only, you will study with me that there is another stain called rhodamine. And I'm teaching you today, so in pathology it will be a test for you. What is rhodamine versus what is rhodamine? Dhyan se, just one alphabet changes and the story changes. When I say rhodamine, like I had said auramine. So auramine, rhodamine, they are dyes for which bacteria? For Mycobacterium TB. Auramine, rhodamine for Mycobacterium TB. If I write rhodamine, that is the special stain for anyone who has read little pathology earlier. That is the special stain for copper. So in the next class of patho, when I will teach you stains of copper, you will study with me about rhodanine. Okay, rhodanine copper, but rhodamine is Mycobacterium TB. Okay, what is the fluorescent stain that we use for malaria? That is going to be acridin orange. It is going to be acridin orange. What is the fluorescent stain for fungus? It is going to be calcoflower white. How I have learnt it, what I believe you will need some mnemonics because you will forget it in a short note. MTB के लिए तो औरामीन रोडामीन okay with everyone, you will remember it, done. Fungus के लिए I have learnt flower. For fungus it is going to be flower, and if I read carefully this sounds like fluorescent. It is the fluorescent dye for fungus. Calco flower white नहीं बोलते, let's call it calcofluor white so that you remember it's a fluorescent dye for fungus. Now, how many of you have heard of this was all micro. I am not teaching you only micro, I am teaching you patho also. So I will always integrate the subjects so that it will help you in every exam. Have you heard in pathology about a technique called flow cytometry? In patho, have you heard of flow cytometry? Flow cytometry is a technique which I use in testing blood samples. In hematology means in blood. Acha, even if you've not heard, have you all heard of something called B cell, T cell? They have some CD markers that this cell is CD1 positive, this cell is CD5 positive, CD15 positive, CD30 positive. You must be wondering what in pathology keeps on going CD10 positive, CD30 positive. Who calculates the CD markers? Which test tells me about the CD markers? It is the flow cytometry. In flow cytometry also you use a fluorescent dye. The name of that fluorescent dye is going to be FITC. FITC stands for fluoroisothiocyanate. I'll repeat it for you. It is FITC, means fluoroisothiocyanate. Together we make it FITC. So now everywhere that you have to study any kind of fluorescent dye, you are done. Jaldi say, without looking at it, you will tell me. What are the dyes for TB? MTB. So for MTB it was auramine, rhodamine. What is the dye for malaria? It is going to be acridin orange. What is the dye for fungus? It is going to be flower, calcoflower white. What is the dye for flow cytometry? For flow cytometry it is FITC, fluoroisothiocyanate. And if I give you this ever as an MCQ based question, will you be able to identify that which microscope is this? This is a fluorescent microscope because ma'am, ultraviolet light was there, dichroic mirror was there, so fluorescent microscope. Okay, but now I want to tell you, can you give me a yes or no, everyone? Wake up and give me a yes or no. Will the fluorescence, that green color, come only because I have added some color? That is why the green color is coming. Apne aap thodi. Ma'am, you have added auramine, rhodamine, acridin orange, something you have added. That is why you are getting this kind of color. But what if you don't add anything? What are the things that can shine on their own? Means, for example, if you say this, let's assume this is one molecule. Okay, this camera cover of mine today is one molecule. I say I have not added any dye onto this, but it is becoming totally green. It is shining. Apne aap it is shining. Have you seen there are some things called fireflies? They are also in Hindi, they are also known as jugnu. They tend to these flies which if you see them in the dark, they emit light. Apne aap se, they emit light. Similarly, if I say, if this is a normal any bacteria, I have not put any color on it, but apne aap it is showing me fluorescence. What are all the things that are autofluorescence? On their own they can show fluorescence. So this is not a bacteria, it's actually a parasite by the name of Cyclospora and Isospora. The spora brothers show autofluorescence. Cyclospora and Isospora. If you've not read about it, never mind, you will be studying it with me under parasitology. Isospora, cyclospora, koi color nahi add karna, apne aap you will see they are shining. Second, formalin is autofluorescence. Where have you seen, have you ever witnessed formalin as an undergraduate in your first year, second year also up till now? First year mein you entered into the dissection hall, anatomy dissection hall, your eyes started burning because all the cadavers were stored in formalin. Your eyes were burning because of formalin. Yes. In second year when you entered the pathology lab, then also your eyes were burning because in pathology lab also all the specimens are stored in formalin. All human body parts are stored in formalin, and formalin is autofluorescent. One more molecule that is autofluorescent which you study in biochemistry is NADPH. If you guys remember in the last year biochem you studied in first year, you must have read one pathway called G6PD pathway. Glucose 6-phosphate converts to 6-phosphogluconate, NADPH is formed. That NADPH which is forming in your body, that can also shine on its own. So they ask you a question, and this could go in a Viva voice question. So towards your Viva, they will say, what are the 3 things which are autofluorescent? And I have seen most students stop at cyclospora, isospora. If you ever tell formalin and NADPH, examiner is not probably going to take your Viva thereafter, not out of anger, but because he would be so impressed that you know everything from path to biochem to micro, everything that is autofluorescent. Now let's move forward. We are left with some minor kind of microscopes that you have to know. One is a phase contrast microscope. So you will say, what is the phase that you are trying to create a contrast? For example, if I have a cell like this, I will draw one cell. What is inside every cell? It is going to be a nucleus. Inside every cell, there is going to be a nucleus. So ma'am, along with that, every cell will have some granules, some cytoplasm, so many ribosomes, so many things every cell has. Can you tell me from your physics knowledge, will the refractive index of the nucleus and the refractive index of the ribosomes and the mitochondria and the cell membrane be the same? Sabka refractive index same hota hai? No, of course not. No two things have the same refractive index. So if I want to look at the internal details of a cell, if I want to look at the internal details of the cell, then ma'am, you will have to play around with refractive index. And that is because there are going to be different refractive index in everything. So I will have to create that difference in my microscope only. So phase contrast, that contrast that you are creating is between the different refractive index. How do I create that? You know, I will be able to see this ribosome also, I will be able to see the mitochondria also, I will be able to see the nucleus also. I'm trying to create that difference that I can see alag refractive index. That is because I have put 2 structures inside. I take a normal microscope, light microscope, but I put 2 more structures in it. One is known as the condenser. I remember ma'am, the condenser, this time the condenser is in the shape of a ring. Condenser annulus is added. And second, near my eyes, there is something called as a phase plate that is added. So 2 things. One is added right at the bottom, that is going to be condenser annulus. One is added right next to my eyes, that is a phase plate. They will make sure that you can see the internal details, while you can see everything with all different refractive indices. Whether the refractive index of this is 1 and this is something else, you will still be able to see them as separate. So simple. When you want to see structures with different refractive indices, what microscope do you use? Phase contrast microscope. Micro mein ho sakta hai na पूछे, patho mein this will be asked. I am teaching you microscopes together. Wherever the short note comes, you write the same thing. Now let's move on to the next one, and that is going to be the electron microscope. Before I start, the last microscope, electron microscope was given by which scientist? Oh no, I think you ended up seeing. But anyway, last class I taught you scientists, right? And I taught you the scientists for light microscope and for electron microscope. It was L for L and E for E. Light microscope was by Leeuwenhoek, Anton von Leeuwenhoek, and E for E. So electron microscope was by Ernst Ruska. Last class we had studied this in the scientists. Now the difference is, I look at this microscope and ma'am, it's not looking like a microscope. It looks like that spaceship, a spaceship or a missile that it is ready to be launched in space. That is how it looks like. Why is it looking like this? Why is it enclosed from everywhere? There is no opening only. They have closed it. They are trying to create a vacuum inside it. Reason for creating a vacuum is that I am not using any light till now. Can you just revise the lights with me? What was the source of light in light microscope? Light microscope में transmitted light था. What was the source of light in dark field microscope? It was reflected light. What was the light in fluorescent microscope? Ultraviolet light. I was only using light. Now I'm not using light. Now I am using a beam of electrons. And electrons are not going to work in the air like this. Air will cancel out electrons. If you want electrons to work, you will have to create a vacuum. And that is what I have done over here. These 2 terms I will come back to later. But I want to simply ask you that if you ever have to do a difference between a light microscope and an electron microscope. This difference between can come in your exam as a short note or a difference between question also. So first, I will now tell you in a very step-by-step manner. I have written it also in a very step-by-step manner. Before that, I just want to ask you, which is a better microscope? If you want to see the tiniest of details, full detail, which has a higher resolution, of course it is going to be electron microscope, has a higher resolution. Ma'am, you had told us it was 0.2 to 0.5 nanometers, whereas light microscope was 0.2 microns. Did I tell you resolution in the beginning? So this you know. Now let's study. This first I will teach you from pathology point of view, then from micro. Let's assume you have a patient who has undergone a kidney biopsy, renal biopsy for whatever ABC disease. Kidney biopsy hua. Two tissues, 1 centimeter tissues were taken out from the kidney. Two tissues were taken out. One has to be sent for light microscope. One has to be viewed under electron microscope. You have today become a urologist, a neurosurgery department head you have become. You have taken out 1 centimeter ke do pieces. Two pieces of kidney biopsy you have taken out. You said that you tell the intern that this 1 centimeter has to be sent to be viewed under light microscope. So your intern is looking at you, how should I send it to the lab? You will say put it inside a bottle which has a liquid. What is the liquid that you will place it in? So you will take a bottle and in that you will place that kidney biopsy. But that liquid that will be filled in that bottle is 10% neutral buffered formalin. Basically, that is why your eyes are always burning in a pathology lab, because all the samples are come dipped in formalin. That is known as 10% NBF, neutral buffered formalin. Next, you say, okay, now the next 1 centimeter has to be sent for electron microscope. Now, which container should you put it in? Now, the fixative you will put it in is 2 to 2.5% glutaraldehyde. This is going to be your job later on as an intern. You will be standing in the OT. If the surgeon says, I've taken out this biopsy, send it for a light microscope. You should know na that which dabba should I place it in. You shouldn't be looking at others. So to send it for light microscope, you put it in formalin solution. To send it for electron microscope, you put it in glutaraldehyde solution. Now you've sent it to the lab. Intern ka kaam khatam. Now let's go to the lab and let's see what the lab people will do. Lab people will say, okay, thank you for sending it to me in a solution, but they will now throw the solution. They will take this biopsy out and they will now put it somewhere else. They can't always let it stay in formalin forever and let everyone's eyes burn. Not possible. So what will they do? And I've shown this exactly to you. Can you see what has been created over here? This is a like candle wax. Actually, that is what we do in a path lab. We take candle wax, and this was the endometrial biopsy or a prostate biopsy or a brain biopsy, a kidney biopsy. We have actually put it inside paraffin wax forever, and now I can store it forever. 5 years, 7 years. Usually we say na human tissue will degenerate, sad jayega, it will degenerate. Inside this it will not. Now it is for eternity, you can say. So what have I done? I have done embedding inside paraffin wax. So they will ask you, I have done embedding inside paraffin wax for light microscope, whereas for electron microscope you can't use paraffin wax with electrons. For electron microscope you do it inside a solution called resin. So when they are asking you, that is for embedding, what do you use in a light microscope? We are going to use paraffin wax. What do we use in electron microscope? We use resin. Finally, very good ma'am, you are ready. Now you have stored it for eternity. Actually, next time you go to a path lab, you only see glass slide and come back. All of us have done that in our second year. But next time, you will see there will be a big room where you will only see lots of paraffin wax and you will be wondering, ye log candle wax ke saath kya karte hain? That is where they embed all the biopsies, and every biopsy is put in a separate candle wax, paraffin wax block. Now what can you do? Like all of you in your childhood, on Diwali especially, when you light candles, do you like scraping the wax later on, next day? Have your parents ever made you do that? That now all the candles have blown away, so now all the wax is on the floor, so now you sit and scrape the wax. Yes, can you do? And how do you scrape the wax? Either you use a sharp object or a blade or a knife, and that is when you scrape the wax off from all the railings. Do you do that, or are you not getting my point? I'm sure all of you have done that. Anyone living in India has done scraping of candle wax off after Diwali, right? So that is exactly what I will do. Now, if I want to look at this under the microscope, I can't look at wax under the microscope. Na? I will take a blade and I will cut one slice out of it. and this is what I will put on my slide. Same way from resin, I will put it on the slide. So you will ask me, ma'am, what slide is used? You know it. What is the slide that you are using in your labs for light microscope? For light microscope, we are using glass slides. But electrons don't work with glass. For electron microscope, you will have to use metallic slides, which are made up of copper. See, you need to understand you're working with electrons. Electrons don't work similar to how light will work. So for electrons, everything is different. Finally, you are done. What is the source? For a light microscope, you will be using light, transmitted light. And for electron microscope, you will be using electrons. For electrons, make sure that the medium has vacuum. Whereas for light microscope, you keep that microscope here on my table, air mein chalega. There is not a problem. Finally, your entire story is done. So now listen to the story. Let's say you are doing a light microscopy. So what is step by step you will do? First, you will put it in a solution called 10% neutral buffered formalin. After that, you will embed it in paraffin wax. Then you will cut a slice. You will put it on a glass slide. Then you will look at it under the microscope. The microscope will have transmitted light, and the microscope will be kept over here on this table in air. But if you are going to look at it under electron microscope, then send it in fixative glutaraldehyde, put it inside resin, cut a slice, and you have to put it not on a glass slide, on a metal slide. Thereafter, you will look at it under microscope, which will transmit electrons, and then you will have to keep that microscope like a spaceship. You will keep it over here, which is known as a electron microscope. Full spaceship, you have created a vacuum. Okay, so we are finally done. Now we come to advanced things. This is written in your textbook. What is shadow casting? You will say, ma'am, everywhere you were adding colors. Fluorescent color, blue color, pink color, kuch you were adding. In electron microscope, you're not adding anything. Do you guys remember I showed you electron microscope in patho also? And I told you that you always get black and white photos. You remember that first photo of patho that I taught you? That electron microscope gives you black and white picture. And today you can ask me, why, ma'am? Why electron microscope always gives me black and white pictures, whereas all the others give me blue, pink, yellow, colorful pictures? Because in electron microscope, I have not added any color. With electrons, I can't add blue, pink, yellow colors. They will not work. I do something called shadow casting. Shadow casting means I add heavy metals. Heavy metals create a shadow. That is why the black and white photo comes of electron microscope. I have added heavy metals. That is known as shadow casting. Next, we realized, I don't know how many of you noticed, look at the size of the tissue when it was in paraffin wax. It was looking pretty big, 2-3 centimeters. The same tissue, when I put it in resin for electron microscope, does the tissue look very small, as if it has shrunken? Actually, we realized that is what was happening. For electron microscope, when I was putting my tissue into resin, the tissue was shrinking. It was becoming so small, so tiny, so shrunken that cells ही नहीं दिख रहे थे. Later on, I could not see cells under the microscope. Then they said, this is not good. If you shrink it to such small levels, what will you see under the microscope? All your cells have been destroyed and distorted. Then we started doing a technique known as freeze etching. Freeze etching is known as freezing the tissue. Freezing the tissue. And they said, don't put it in resin, because resin is making your tissue go very, very shrunken up. Put it in some deep freezer. It's a very criminal thing for me to be discussing, but you must have seen in all those crime stories that so-and-so's husband or so-and-so's girlfriend or boyfriend then killed or murdered his or her partner and then cut his body parts into so many pieces and had stored it in the deep freezer. You've heard those criminal stories that keep coming up, which are so disturbing. Why is it that these people who are doing all these things are storing always the stories that they're storing the body parts in deep freezers only? Because the metabolism, whatever cellular degradation has to happen, will not happen because of very low temperature. Secondly, the body parts start shrinking. That is what you are doing here also. You are trying to shrink the tissue, but not that much that resin was doing. You are still shrinking the tissue, but itna shrink kara that you can look at it under the microscope. So you are freezing the tissue prior, and that is known as freeze etching. Before looking at it under the electron microscope, now we have two techniques. For black and white photos, we add heavy metals, that is known as shadow casting. Second, instead of resin, we prefer to freeze the tissue. That is a better result, and that is known as freeze etching. I hope that is okay, because now I tell you, because you guys have been so good with me, you will tell me electron microscope is of two types. That is known as TEM and SEM. These are the two electron microscopes. So what is TEM? TEM stands for transmission electron microscope and SEM stands for scanning electron microscope. Okay, TEM and SEM. Now listen to the names very carefully. What is the principle? You will say, ma'am, electron microscope. In both of them, you are going to use electrons. This also has electrons, this also has electrons. But in transmission, what do you understand? The electrons, if this is the object, if this is the bacteria you want to see, transmission, electrons will go through, this white pencil is electrons. Electrons will go through and through like this, they will get transmitted. So can I say what kind of electrons am I using? I am using transmitted electrons. Whereas in scanning electron microscope, you will say, ma'am, if this is the bacteria, you just want to, जैसे paper scanning होता है ऊपर ऊपर से. So when electrons will come and they will scatter, they will reflect, because you just want to see the surface, you don't want to go inside and see everything. So can I say when I'm using scattered electrons, I'm trying to do scanning electron microscope? You logically tell me in which of them, in see transmission में क्या है, electrons are going through and through the bacteria. Scanning में क्या है, electrons are just scanning, superficial. In which of them will you be able to see the internal details of a cell? The electrons going through and through and through, ma'am, then I will be able to see the internal details of the cell. So in transmission electron microscope, I will be able to get the internal details of the cell, whereas in scanning, I'm only looking at the surface, that's it. So where do you get only the superficial details of the cell? That is going to be scanning electron microscopes. Have you understood? Don't mug up these differences the way they are written. You actually have logic behind them. Next question. This is something you will have to mug up. Transmitted electron microscope gives you, because see, it's going through and through. It can only show you 2 dimensions, outside, inside, bus, 2 dimension. Scanning on the other hand on the surface will be able to show you the full 3D view. So scanning electron microscope is going to show you a 3-dimensional view. Next. Which out of these do you think is going to, if I say you have 5 minutes to do electron microscopy, let me ask your logic. You have 5 minutes to do electron microscopy. In which out of these 2 electron microscopes will you be able to cover greater surface of the bacteria, that you can see the bacteria from this end to this end? You will be able to cover greater sample in lesser time. In which of them? Ma'am, superficial देखना तो easy है, जल्दी you will see from here to here. So can I say you will be able to cover a greater amount of sample viewed in lesser time? Greater amount of sample viewed in lesser time. Do you all agree with me? Versus if I'm using transmission, ma'am, all the electrons are going through and through, पूरा detail में पढ़ाई चल रहा है, detail में the study is going on. So you will say lesser amount of sample is viewed in the same time. Lesser amount of sample is viewed. Do you all agree? It's something like right now what are you doing with me? Full course study, ma'am. Right now we are doing transmission electron microscope. We are spending 1 hour only doing microscopes. I'm only studying 1 hour. I'm doing very little topic. Why? Because I'm going into every little detail and clearing my concept. But when I'll be 1 hour away from the exam, I will start doing superficial, scan the pages, jaldi. Then I will cover microscopes in 5 minutes. I will do greater amount in lesser time. So scanning is like your rapid revision, and transmission is these Prof Edge classes where you are going into the innermost details and studying everything in great extent. Have we understood all the microscopes? Everyone, are you ready for some questions? I've only got A. I want you to cover the main videos so that you don't have to view them. Second, अपना NEET PG का तैयारी साथ साथ चलेगा. University and NEET PG hand in hand. So what is the maximum magnification that can be attained in a light microscope? I think we all know it. Answer is going to be 1000x. We discussed it so many times. Perfect. A microscope that exposes the specimens to ultraviolet light and forms an image that is emitted at a different wavelength. Ma'am, ultraviolet light was used only in fluorescent microscope, and the next sentence says you had taught us that shorter wavelength is converted into a longer wavelength, and that is also right because it is emitted at a different wavelength. Yes, fluorescent microscope, very good. Which of the following structures are required in the microscope for taking this kind of an image? This image that I can see is a fluorescent microscope image, right? In fluorescent microscope, what do you need? Do you need a dark field condenser? No, that was in dark field microscope. Cathode ray tube तो कहीं भी नहीं चाहिए. Phase plate I would need is phase contrast microscope. For fluorescent microscope, I would be needing dichroic mirror. Very good. Two things needed. One is ultraviolet light needed. Second thing that is needed is going to be a dichroic mirror. Perfect, done. So we had a 40-minute target, we exceeded by 20 minutes, but that's okay, but we finished it. That is what I say, time boxing. You will make a target, and as human tendency goes, you will always exceed that target. But if you don't meet that target, you will never finish it. You'll keep contemplating and thinking, will I be able to do it or not? You think of 40 minutes, you take 50 minutes, but you'll finish it. Better than not starting. That is how time boxing works. So now the next 1 hour leaves us with bacterial anatomy for the day. This is bacteria is what I'll start with, bacteriology. So I'm giving you all the basic anatomical structures here in general microbiology, then individual bacteria we will do later on. So right now what I realized, is magnification same for TEM and SEM? Dr. Anish. Dr. Anish, no. Nowadays we have many advanced microscope. In fact, a new microscope has come up called high voltage electron microscope. That everything has a different magnification. That I cannot give you in writing, because magnification will always depend on from which company it has been made and what lens system are they using. Like for light microscope, I told you it is standard 4, 10, 40, 100. But for electron microscopes, there are many foreign brands also which are manufacturing electron microscopes, and everyone has a different set of lenses that they use. So different types of magnifications are available, and this is no textbook thing that you have to learn that only 100 times or 1000 times it will be magnified. That depends on who has made the microscope, which company, which manufacturer. But the magnification is not the same. Okay, coming back. So yeah, bacterial anatomy. What do I see? I see outermost, don't write anything. Outermost is something called a capsule. Thereafter, ma'am, I can see there is going to be a cell wall. Then there is some membrane, dotted membrane you have made. Then there is a cell membrane. Then it must be the nucleus and some other dot. Then I can see one thread called flagella coming out, and then I can see short structures called fimbriae coming out. Bas ye padna in 1 hour. This is your next 1 hour. So I start off with what? I start off with the outermost layer, and that is known as a capsule. Some people call it a capsule and some people call it a slime layer. Firstly, capsule kya hota hai? Capsule is something which is very well demarcated, like I have made over here. It is a rigid covering of the bacteria. A rigid covering of the bacteria is going to be a capsule. Instead of capsule, sometimes there is a slime layer that is present. Slime layer is very loose. It's do you guys know what is slime? Slime एक बच्चों का toy आता है like this. It's something which is very loose and you can easily pull it apart. The slime layer actually looks like that. Slime layer is very loose and undemarcated. Ma'am, do they mean the same? Yeah. Are they made of the same material? Yes. It's just how they appear. Some people appear very strict and some people appear very lenient. Or for example, your parents in front of you, they are like capsules. They are very rigid and they say you will follow our rules. But your same parents in front of their friends, they become slime layers. They become very loose and very uninhibited and very chilled out. That is how same person, different situation, different appearances. Same way. Capsule or slime layer hai same, but it looks different. Capsule is going to be very rigid. Slime layer is going to be very loose. So any one will be present. Either a bacteria will have a capsule or a bacteria will have a loose wala slime layer. Is there any bacteria which can have both? Viva question. It is Streptococcus salivarius. Streptococcus salivarius can have both the capsule and the slime layer present in it. So does every bacteria have a capsule or a slime layer? No. Obviously, the ones which have a capsule, they are going to be capsulated bacteria. Some people call it capsulated or encapsulated, same cheese. The ones which don't have a capsule are known as unencapsulated bacteria. So there are two options, capsulated bacteria and unencapsulated bacteria. What is the advantage that these capsulated bacteria have? If a bacteria has a capsule, it will act as a virulence factor, means when it will enter into my body. Just imagine when a bacteria enters into our body, which is that cell which always goes and eats everything. That one cell that is always hungry is macrophage. Macrophage goes and eats everything, right? So when bacteria will come into my body, I would also want to eat it. But now who will protect the bacteria from phagocytosis? Capsule. Capsule is like a shield. that you cannot eat me. I am a capsule. I am very rigid. I am very well rigid and demarcated. You cannot eat me. So capsule is like a virulence factor. And now, right now, I don't want you to learn it because abhi to ma'am, we don't know names of bacteria. But if anyone asks you which are all the capsulated organisms, just read through once. You know, there are so many lists in microbiology that we'll have. I will always make you learn the lists in two ways. One, I will give you a promo, a trailer. Like today is a trailer. अभी कोई bacteria का नाम नहीं पढ़ा. I will say एक बार पढ़ लो. Out of 10 you will remember 2, 3, 4 at the max. Again, when I will start bacteriology, when I will finish all these bacteria, I will then sit and make you learn it again. So first, for every list in micro, you will get a trailer, a promo, and then the final movie, and then you will have to revise it with me. 3 times you do, apne aap list yaad hoga. So if I ask you which are all the capsulated bacteria, they are learnt as pretty nice capsules. Pretty means all the P's. I can see lot of P's written over here. Now I'll give you a way of learning this. Have you heard of Clostridium? So many Clostridium are there. At least all of you have heard of Clostridium tetani, जो tetanus कराता है. So ma'am, I have heard of Clostridium tetani. So Clostridium species में does tetani have a capsule? No. Which is the Clostridium that will have a capsule? P वाला Clostridium. Perfringens has a capsule. Okay. Vibrio. All of you have heard of Vibrio cholera, which causes cholera, diarrhea. Does Vibrio cholera have a capsule? No. Which Vibrio has a capsule? Vibrio parahemolyticus. Which Bordetella has a capsule? Bordetella pertussis, which causes whooping cough, pertussis. Which Klebsiella has a capsule? Klebsiella pneumoniae. Which Streptococcus has a capsule? Streptococcus pneumoniae. So later on, you will study 10 types of Streptococcus, 10 types of Klebsiella, 10 type of Bordetella, but you will know all the P family members. They are the ones which are going to have the capsules. Abhi ke liye, this is how you learn it. Streptococcus pneumoniae, Klebsiella pneumoniae, Bordetella pertussis, Vibrio parahemolyticus, Clostridium perfringens and Yersinia pestis. Her family member ka P member is going to have a capsule. Next, the way you read nice. Nice sounds like Neisseria. Nice is Neisseria meningitidis or meningococcus. ये meningitis कराता है. So that is also going to have a capsule. Nice हो गया. I say I'm sure influenza तो सबने सुना हुआ है. Hemophilus influenza, cold, common cold, you've heard of it. Okay, let's go. Capsule. C for any fungus. There is a fungus called Cryptococcus. This has a capsule. Okay, anyone, what does Cryptococcus cause? Cryptococcus causes meningitis. Okay, ma'am, you said Neisseria meningitidis also causes meningitis. And H influenza, this though common man can also tell it causes influenza. What other that influenza does it cause? It is also going to cause meningitis. So do you realize all these bacteria which go to the brain, they must be damn strong. That is why they go to the brain. And what has made them this strong? The capsule. The capsule has made them strong. Hence they go to the brain and they cause meningitis. See the logic behind it. Capsule is giving strength to a bacteria. So all these meningitis bacteria are going to have a capsule. Okay, C for Cryptococcus, A for Bacillus anthracis and Staphylococcus aureus. Now let me tell you something. What are capsules made up of? अभी बस एक बार पढ़ा. Don't force your brain कि मुझे ये list याद होना ही चाहिए. You will automatically learn with me when we do bacteriology. All capsules, what are they made up of? All capsules are made up of polysaccharide. This is what you should have asked me. कि ma'am, slime layer, capsule, what are they made up of? They are made up of polysaccharides. However, the rule is that an exception will always be there. If a rule is there, exception is always there. Except, I could see in front of Bacillus anthracis, ma'am, you have written polypeptide. Instead of Yersinia pestis, you have written peptide. They are not made up of polysaccharide. Please remember Bacillus anthracis and Yersinia pestis. What are they made up of? They are made up of polypeptide. Is that okay with everyone? Now repeat. ये नहीं आता. Pass fail वाले questions जो होते हैं ना, मतलब दोस्तों, you have to repeat 2-3 times with me. All capsules are made up of polysaccharide, but Bacillus anthracis and Yersinia pestis are made up of polypeptide. Okay, all capsules are proper capsules, visible capsules. Which of them is a microcapsule? And that is going to be Staphylococcus aureus. It is very tiny, sometimes present, sometimes not. Capsulated organisms seem to be done for you. So now if I ask you the next question, that what is the demonstration? अब आप देखोगे कैसे? how will you look at it under the microscope? Or forget microscope, what are the tests that you can do? You want to find out कि मैम आपने मुझे बताया था pretty nice capsule. How did you get to know that these are the ones which have a capsule? I did two tests. One is known as Macfadyen reaction. Second is known as Quelling reaction. I've written one bacteria in front of it. Tell me, does Bacillus anthracis have a capsule? Yes. Does pneumococcus, you said ma'am P wale to everyone had a capsule. These have a capsule. Fine. So what is Macfadyen reaction? In Macfadyen reaction, I'm going to take a bacteria. I am assuming, chalo maan lete hain, that the bacteria has a capsule. Okay, I have a capsule. What do I do? To this, I am going to add a purple color dye that is known as polychrome methylene blue. What have you done? You are a microbiologist. You took a bacteria. Right now you don't know capsule hai nahi hai pata nahi ma'am. I took one bacteria. To that bacteria I have added polychrome methylene blue. If I see that, okay, I'll again draw. If I see this is a bacteria and now there is some purple color that has formed around it. If the bacteria is showing me a purple color, you will say, oh, that can only come if a capsule would have been there. If capsule wouldn't have been there, then this purple color would have not come. If purple color has come, this means there was a capsule. This is known as Macfadyen reaction. How I have learnt it? M for Macfadyen reaction. What is the stain that you have to add? Methylene blue. In Macfadyen reaction, repeat with me, what color do I add? Methylene blue. If you get a purple color, this means capsule is present. What is the second test that you do? Quelling reaction or Neufeld reaction. So let's assume again this is a bacteria, and for the time being let's say that it has a capsule around it. What is capsule? You will say, ma'am, capsule is nothing but antigen. Every bacteria that is carrying a capsule, indirectly it is going to carry an antigen in the capsule. Okay, so now that obviously means that in my lab, if I add any antisera, when I say antisera, can I in other words also say antibody? Tell me something guys from your basic, even if you've not read anything, whenever there is an antigen and I end up adding an antibody, तो क्या antigen antibody reaction होगा? Of course ma'am, there is going to be a reaction, which means that antigen के ऊपर I have added an antibody. As soon as the reaction will occur between them, this capsule will become like a big balloon. This reaction will result in swelling of the capsule. Why? Because antigen antibody reaction occurred. How I have learnt it again is Quelling may what happens? Swelling. So now what are the two ways of identifying a capsule? Lastly, what I told you right now, we did transmission electron microscope. Pura transmit dissect karke detail pada. Now I will show you scanning electron microscope. How to scan this page in 1 minute, or not 1 minute, 10 seconds. Macfadyen reaction, what do you add? M for M. I've added methylene blue. If I get a purple color, capsule is there. Quelling reaction, I see for swelling or swelling. Antigen antibody reaction hua, swelling will happen. This means Quelling reaction is positive. Okay with everyone, you have done the scanning and capsule story is done. Now, telling you in detail. I told you capsule was very tough, rigid. Then a more loose version of it was slime layer. Now further modification of slime layer is known as biofilm. This is the most trending topic. I mean, if you have professors in your college who are too much into updates, very few medical colleges have academically teachers who are very up to date with things, you know, who are very fond of teaching academically. And if you have any such teacher who is going to set your paper, this is the short note that you will be getting. What is a biofilm? We saw that capsule tha, or that loose slime layer was there. In some bacteria, the slime layer modified into a biofilm. Is it good for the bacteria, bad for the bacteria? Let me read. What is a biofilm? Biofilm, ma'am, makes the bacteria antibiotic resistant. I'm feeling scared. Okay, bacteria is getting superpowers. Bacteria, because of this biofilm, will become antibiotic resistant. I will keep giving antibiotics and the bacteria will not get killed. I'm feeling very scared. So is biofilm a superpower for the bacteria? Yes. What else? It is going to make it antiphagocytic. Means I am trying to eat it, phagocytose it. Bacteria says, no, I will not let you do that. I have biofilm. Third, it adheres, sticks to my body. It is forever adherent. I can't take it out of my body. So I have realized that biofilm has made it antibiotic resistant, adherent and antiphagocytic. I should be very, very scared that no bacteria should ever make a biofilm. If a bacteria makes biofilm, then human beings are going to suffer a lot, and that is what we do. I will give you some examples of bacteria which end up making a biofilm. There is a bacteria which goes by the name of Streptococcus mutans. Anyone has any idea what Streptococcus mutans bacteria does? When I'll tell you, you'll be like, is it? When I got to know, I was very amazed that we say something every day and we don't know the meaning of it only. All of you have heard of dental caries. Some of you might be having dental caries. It is a very common thing. Dant mein keeda lagna is how the common man calls it, right? So dental caries is such a common thing, ma'am. We have it, or family friends have it. Have you ever wondered what is dental caries, or are you also like all of us? Till a very long time, I also used to think it is just that the teeth have, जैसे our parents told us, दांत सड़ गए, because you used to eat a lot of sweet in the night, you never used to brush in the night. All of that reasons they used to give, but actually it was a bacteria which was coming and sticking, and that is Streptococcus mutans, which is responsible for dental caries. Streptococcus mutans, how I've learned is mutans reminds me of mouth, so it is going to cause dental caries. And now I understand why does it come and stick to my teeth? Because it's got a biofilm. Biofilm gives it superpowers to go and stick somewhere. This is example number 1. Example number 2 is going to be, right now you might not understand the importance of it. Later on with me in medicine, in path, you will understand. Staphylococcus epidermidis. Staphylococcus, a name you might feel epidermidis, ma'am, epidermis, yes, derma se related hoga, no. This causes something called as prosthetic heart valve endocarditis. Prosthetic heart valve endocarditis. I think basic meaning you know. Do you know heart valves? Yes, ma'am. Mitral valve, tricuspid valve, pulmonary valve, aortic valve. I know heart valves. They are having itis, matlab inflammation. Okay. What is prosthetic? People who have undergone a heart valve transplant. They don't have original, hamari tarah, original heart valves. They have got some surgery and got an artificial heart valve put into their body. Their artificial heart valve can get infected with which bacteria? With Staphylococcus epidermidis. Why? Because it is having biofilm. So Staph epidermidis and Streptococcus mutans, they are famous for biofilm, which will give them superpowers. What are the 3 superpowers? Triple A superpowers milega unko. Antibiotic resistant, adherence, and anti-phagocytic. Okay, so I think I'm done with the very first layer. Whatever I keep teaching you, we will keep labeling. Number 1 layer, which was on the outermost, that is capsule, or you want to call it slime layer, that is settled for us. Now let's move further. What beyond it, guys? What is the next layer that I have? That is going to be a cell wall. Now when I say cell wall, did you wonder when I was teaching you gram staining in the last class, I always used to tell you gram positive bacteria and gram negative bacteria. Did you wonder why do some bacteria become gram positive and some bacteria become gram negative? Because they have a cell wall which is gram positive and gram negative. Now before that, all of you, can you tell me gram staining ka protocol kya tha? I'm just asking you the mnemonic. Come in and stain. Do you remember? Come in and stain. Number 1, you were adding crystal violet. Second, you were adding, yes, iodine. Third, you were adding some alcohol or acetone. And lastly, you were adding saffron in red. And I told you, gram positive organisms will become purple in color. Gram negative organisms will become red or pink in color. Very good. So only you will see, that's why I say every class will have continuity with the previous class. And if you cannot revise 1 or 2 classes, I mean path micro pharma मिला के 3 classes in a month, तो आगे जाके तो you will have 9 to 10 a month. So you need to speed up with us gradually like that. Otherwise we could have bombarded you with 30 days, 30 classes and finished off path. It was so easy for me. Every day 3 classes, 90 hours patho khatam. But that would have not gone. Infiltration into your brain wouldn't have happened. Precisely what we want you to do. So don't come without revising the previous session, not for me, for yourself. You will benefit if you revise the previous class. Now you will understand why was gram positive purple and why was gram negative red. You will now understand the reason behind it, because their cell wall looks different. Let me first show you the cell wall. Don't write anything, just idea le lo kya hai. Gram positive cell wall, gram negative cell wall. I see one difference. This blue color layer is very, very thick in gram positive. What is this blue color layer? It is peptidoglycan. So much of peptidoglycan is present in gram positive. Very little peptidoglycan is present over here in gram negative. एक difference तो मुझे दिख गया. What else? I can see some filaments going over here called teichoic acid. Ma'am, I can see teichoic acid only in gram positive. I don't see any teichoic acid in gram negative. Okay. So whatever you're telling me, why don't I write it? When I'm talking about gram positive bacteria, what is the first thick layer that you saw? You saw lots and lots of peptidoglycan. Also, what thing did you see? You saw lots of teichoic acid. अभी तक मैम इतना समझ आया. Details I will go into what basic I understood. Let me see one more picture. I am looking at it from a very superficial point of view. You said okay, all this is peptidoglycan, but why is it made in the form of these boxes? Because every cell wall is going to have boxes. The name of those boxes are going to be, can you see, instead of boxes I have made circles. These are known as NAM and NAG. So first I will explain to you what is NAM and NAG, and then we will go into the detail. Anyone, I'll write it again for you because otherwise like this it's very tiny font. All these yellow color ones are known as NAM. Any full form for NAM that you want to tell me? N-acetyl muramic acid. N-acetyl muramic acid. Whereas one of this is going to be, the blue one is going to be NAG. NAG is going to be N-acetyl glucosamine. I mean, for NEET PG students, sometimes I say you can skip the full form. But for you guys, university exams, right? So we have NAM, NAG. If you look carefully, NAM, NAG is alternating. N-acetyl muramic acid, N-acetyl glucosamine. ध्यान से देखो, it is going to be yellow, blue, yellow, blue, yellow, blue. Means it is going to be NAM, NAG, NAM, NAG, NAM, NAG. फिर NAM, NAG, NAM, NAG. मैम gram positive में भी NAM, NAG, NAM, NAG चला. I go to gram negative over there also, NAM, NAG, NAM, NAG, NAM, NAG. So ultimately I realize that whether I'm dealing with gram positive or gram negative, NAM, NAG तो same रहेगा. What's the difference? Now look at this carefully. If this is yellow color, one NAM, and this is yellow color, one more NAM, do you see something hanging from these NAMs? 1, 2, 3, 4. 4 proteins. 1, 2, 3, 4. ग्राम पॉजिटिव में तो 4 प्रोटीन हैंग कर रहा है. ग्राम नेगेटिव में ढूंढते हैं मैम. इन दैट ऑल्सो 1, 2, 3, 4 and 1, 2, 3, 4. So how many proteins, whether gram positive or gram negative, how many proteins are attached to NAM? So in gram positive also 4 proteins are attached, and in gram negative also 4 proteins are attached, called tetrapeptide chains. In gram positive they are known as agla. That is how we've learnt it. Means the 4 proteins are alanine, then it is going to be glutamine, then lysine, and then back to alanine. So alanine, glutamine, lysine, alanine. In gram positive, it is going to be agla. A gram positive, a positive person will always move forward in life, will not get stuck to the the past, will always move forward. He will say, what is coming up next? What is agla? So remember, a gram positive person will always ask you, what's coming up next? What's agla? Gram negative mein I realized almost same hi chal raha hai. See, it's the same alanine, it's the same glutamine, ending is also alanine. The only thing is, instead of agla, instead of lysine, it is going to be diaminopimelic acid that is going to be there. If you can learn it, very good. If you can't, then you can actually write it in the exam, and every examiner will accept that in gram positive it is agla. In gram negative, there is replacement of lysine with pimelic acid, वो भी चलेगा. If you don't want to learn diaminopimelic acid, ये amino acid ये remember करना है क्या? या डॉक्टर हिमांशु एक्चुअली नीट पीजी के लिए तो ऐसे स्किप कर दो चलेगा. आई मीन फॉर एंट्रेंस एग्जाम्स, यूनिवर्सिटी एग्जाम में तो लिख के आना पड़ेगा ना, बिकॉज सेल वॉल इज द मोस्ट एक्सपेक्टेड क्वेश्चन आउट ऑफ ऑल ऑफ दिस. सो फॉर यूनिवर्सिटी एग्जाम्स, you know, it has, some people say university exams are easier. I think they're tougher, because at least in MCQ exams the answer is in front of you. Even if you remember kuch to tha pimelic, you will find the answer. But in university exam you will have to write diaminopimelic acid. So then it becomes tougher because you have to learn that entire thing. So gram positive agla and gram negative में only lysine is changed. तो दोनों मैम ultimately दोनों में 4 अभी भी difference क्या है मैम? difference तो नहीं पता लगा. Now I'll show you. You saw 4 proteins agla over here and then 1 agla over here. Agla and agla are connected with how many proteins? Please count and tell me. I can do the counting, but then I don't have to learn. You have to learn. This is agla. I will write agla over here, and this is also agla. Ma'am, agla and agla are connected by 5 proteins, pentapeptide. Gram positive में connection क्या है? 5 proteins. Gram negative में are you seeing any connection? I don't see any connection. That's the final difference that I was waiting for. Out of gram positive and gram negative, which of them is going to have the pentapeptide bridge? Which of them is going to have the pentapeptide bridge? Gram positive has the pentapeptide bridge, means if this is the agla, then this is also the agla. So the 5 proteins combining agla and agla, this is going to be seen only, that's why I called it a bridge. This is seen only in gram positive. तो देख लो अपना mnemonic बन गया. Now if you forget everything, this mnemonic will stay with you. Short note लिख लोगे. In a gram positive organism, which layer is very very thick? Peptidoglycan layer is very thick. What is agla and agla connected by? Ma'am, they are connected by a bridge called a pentapeptide bridge. And what are the proteins which are present? They are going to be teichoic acid. Now let me start over there and asking you that everything is so present in gram positive. What is this poor gram negative doing? Does gram negative have peptidoglycan? Hardly any. Does it have pentapeptide bridge? It doesn't. Does it have teichoic acid? Bichara, it doesn't. So gram negative has nothing, ma'am. God must have given it something. God has given it lipopolysaccharides. That is the speciality of gram negative. ध्यान से देखो, it has lipopolysaccharides. So why don't we just focus on gram negative for some time? बहुत हो गया ग्राम पॉजिटिव का बात, ग्राम नेगेटिव पे फोकस करते हैं. व्हाट डज ग्राम नेगेटिव ऑर्गेनिज्म हैव? लाइपोपॉलीसैकेराइड. सो इफ आई लुक केयरफुली, इट विल हैव लाइपो मतलब सम लिपिड पार्ट and polysaccharide. Two things are certain, it will have lipid part and polysaccharide part. Actually it doesn't have two, it has 3 regions, and these 3 regions are learnt as OPA, very famous. What does gram negative have? It has lipopolysaccharide made up of OPA. What is OPA? Region 1, the first region that is there is made up of O antigen. This is the most variable part. Matlab, what does that mean? Antigen, to ma'am, I've understood, it has an antigen by the name of O. Variable means if this is bacteria 1 and if this is bacteria 2, you will say in both of them, ma'am, both of them are gram negative bacteria. ये दोनों gram negative है. Both of them have OPA. How will you differentiate this from this? I'll say the part which will show the maximum variations will be O antigen. O antigen of this bacteria and O antigen of this bacteria will be different. So the part which is showing the difference between two bacteria, that is the O antigen. So O to समझ आ गया. What is P? अब obviously lipopolysaccharide में P is going to be the polysaccharide. Polysaccharide मिल गया. What is A? Ma'am, lipid part was missing. So lipid A, repeating, O antigen, polysaccharide and lipid A. That is going to be the 3 parts of lipopolysaccharide. So what is this lipid A? Lipid A is where endotoxin will be there. In some time I will be teaching you हर बैक्टीरिया क्या कराता है मैम? फीवर, इन्फेक्शन, that is what it will do to me. Skin infection, bone infection, brain infection, meningitis, every bacteria does that. But some bacteria produce exotoxins, some bacteria produce endotoxin. Toxin हर कोई बनाएगा. Some will produce exotoxin, some will produce endotoxin. Gram negative bacteria usually make endotoxins and they come from lipid A part. Next, lethal effect. The main damaging part is lipid A. Pyrogenicity. What is pyrogenic? Fever कराने वाला ability. So you are like, hang on ma'am, this lipid A is the culprit. Whenever this gram negative bacteria enters my body and we suffer from fever and we feel body aches हो रहा है, and you know sometimes we might have to get hospitalized, all that lethal things are being caused by which region of OPA? It is that lipid A. That will release endotoxin, that will have all the lethal effects, that will cause fever in me. So now let me ask you 3 questions. Question 1, what is more in gram negative organism? Lipopolysaccharide. Question 2, lipopolysaccharide or OPA में से which is the most variable part? Variable part is O antigen. Which is the most lethal part, which is making the toxin, causing fever? The most lethal is L for L, that is lipid A. Whatever I had to teach you, I'm done. Before I show you the table, you will do the diagnosis for me. Guys, ये क्या है? Gram positive cell wall या gram negative? Let me see if you were listening or have you slept off. Gram positive or gram negative? You will say, ma'am, I can see NAM, NAG, NAM, NAG, NAM, NAG, NAM, NAG, वो ठीक है. उसके नीचे अगला भी ठीक है, अगला 4, अगला I can see agla and agla are connected by 1, 2, 3, 4, 5. There is a pentapeptide cross bridge. This is gram positive. Every time I see these 5 connections, gram positive. जो पढ़ा, put it in a table now. Now your differences become so easy. भूल जाओ एग्जाम में कि अगर मैम कुछ भी याद नहीं आया तो पीपीपीटी तो याद आएगा. Gram positive, peptidoglycan will be there, pentapeptides will be there, teichoic acid will be there. So tell me, overall, which had greater peptidoglycan? Gram positive, so it will be almost 100 layer thick, whereas gram negative is very less peptidoglycan. That is what I've written. Composition primarily gram positive has what? It has peptidoglycan, whereas gram negative, I told you predominantly it has lipopolysaccharide. So if it has lipopolysaccharide, they will ask you, देखो ये होता है पेज बढ़ाना. Now if I split it into two, who has more of lipid content? I mean, obviously, lipopolysaccharide jahan pe hai, the lipid content will be more in gram negative, and in gram positive, it is going to be less. This point is very useless. I mean, it's increasing your number of lines. Next, I will show you some other things also before coming back. Where do you think teichoic acid is present? Ma'am, teichoic acid is going to be present in gram positive, okay? Where is lipopolysaccharide present? It is present in gram negative. Where is NAM-NAG present? NAM-NAG present तो दोनों जगह है, but where is the pentapeptide cross bridge present? Gram positive. Where is it absent? Gram negative. यहाँ तक समझ आया? Out of these, no one asked me what is the function of teichoic acid. Teichoic acid is for attachment of the bacteria. When a bacteria comes into, say, your lungs, how will it attach to your lungs? If a bacteria comes to your skin, how will it attach to your skin? For attachment, you have teichoic acid. How I had learnt was teichoic sounds like tikna. So whenever it wants to stick to one place, tikna, okay, whenever it wants to stick to one place, it is going to use teichoic acid. Ma'am, PPPT से हमने ये सारे बना दिए, और क्या बचा? Show me the thing once again. Gram positive, fully covered. Gram negative, you told us lipopolysaccharide. Lipopolysaccharide was made up of OPA. I have seen O for O antigen. I have seen the polysaccharide. I have seen the lethal lipid A done. But now I can see something else. I can see one additional area which is known as outer membrane. Then I can see the little bit of peptidoglycan. And I can see one more space called periplasmic space. कैसे बन रहा है देखो, मुझे एक extra layer दिख रहा है called outer membrane. I can see one more layer which is known as inner membrane, and in between them I can see a periplasmic space. So देखो, God did not give it any peptidoglycan, then God gave it everything else. God gave it lipopolysaccharides, God gave it outer membrane, God gave it inner membrane, God gave it periplasmic space. So now when you will be asked where is the outer membrane present, तो मैम अब जो भी होगा ना, let's give it to this gram negative. उसको बेचारे को कुछ भी नहीं मिला, so we give it to gram negative. Next, if I ask you where is periplasmic space present, this is going to be present in gram negative. So what is the question on periplasmic space that they are going to ask you? This I will ask. This is, I mean, distinction wala question aa jata hai, that if you write this in your university exam, then this is like the topmost detail. Periplasmic space. Sometimes you will see in this periplasmic space one thread is hiding. In some bacteria you will see one thread is hiding. This is, have you all heard of flagella? Flagella तो मैम ऐसे बाहर निकल के आता है like a tail, and the bacteria moves. Locomotion is because of flagella, but over here the tail is not coming out, the flagella is not coming out, the flagella is inside over here, and this is known as an endoflagella. Periplasmic space can have an endoflagella, which is seen in which bacteria? It is seen in Treponema pallidum. Any idea about what does Treponema pallidum, कौन सी family को belong करता है? It belongs to the family of which bacteria? Spirochetes. And for those who don't know, I'm sure you have heard of a disease called syphilis. Syphilis is caused, it's a sexually transmitted disease. Syphilis is caused by Treponema pallidum. It is a spirochete. So you will say, अच्छा मैम वो जो अभी हमने देखा था स्पाइरल वाला ऑर्गेनिज्म, यस, उसका फ्लैजिला कहीं निकलता हुआ दिख रहा था? नहीं दिख रहा था मैम. अभी कौन से माइक्रोस्कोप पे, विच माइक्रोस्कोप डिड यू सी दैट स्पाइरल, वेरी थिन स्पाइरल? आई सेड फॉर वेरी थिन स्ट्रक्चर्स you will make the background dark. On which microscope did you see it? We saw it on a dark field microscope, if you remember. So in that dark field microscope, if I show you again, did I show you any flagella coming out, tail coming out? No, because that flagella is hiding inside. These organisms have something called as endoflagella, and that endoflagella is going to be present in the periplasmic space. That endoflagella is in the periplasmic space. Pura cell wall ho gaya khatam. Now if I want to ask you, how will you see this? थ्योरी में तो पढ़ लिया, लैब में कैसे देखोगे? How do you do the demonstration of cell wall? You have 4 techniques. 3 are for लिखने के लिए. 1 is what I genuinely do in the lab. So in the lab, we have learned it as RMP doctor, because every doctor has to be registered medical practitioner. All of us have to get ourselves registered. So registered medical practitioner doctor. Doctor stands for differential staining. Differential staining means gram stain. Yes ma'am, this I understand, that gram positive, gram negative. This is how I look at a cell wall. Understood. ये तो हम करते हैं. But other than this textbook knowledge, 3 more techniques can be used. Either you react it with an antibody, or you do mechanical rupture, like with needles you can do mechanical rupture, or micro needles you can do micro dissection, and you can do something called plasmolysis. You don't have to know, there will be 5% of microbe, where I will say detail mein mat jao. Don't break your head over it, ratta lagao and forget it, because these techniques will never help you anywhere. So either you react with an antibody, or you try to break it mechanically, so mechanical rupture and micro dissection, plasmolysis and differential staining, that is gram staining. That is how you do the detection of a cell wall. Cell wall also done. Now let's move forward. What if there is no cell wall? What if there is something known as cell wall deficient forms? What are those? Ma'am, cell wall deficient forms are known as L forms. Did I teach you in the last class the scientist who gave you L forms? L forms were given by the scientist Klein-Berger. So why did Klein-Berger, he should have called it Klein-Berger form. Why did he call it L form? What is the purpose of calling it L? Because it was diagnosed at the Lister Institute in London. So this is going to be a bacteria, but that bacteria does not have a cell wall. It is a cell wall deficient form. So you'll ask me 2 things, that ma'am, naturally it doesn't have a cell wall. I mean, by birth only the bacteria doesn't have a cell wall. Ho sakta hai. Or maybe it has a cell wall and then something happened and then the cell wall vanished. Like I'll tell you on a lighter note, all of us, it's not that we were not we were very brainy, right? Till your 10th grade, you must be the topper of your class. In your +1, +2, or in your boards, 11th and 12th, you must have been in the top 5 of your class. Suddenly, when you come to MBBS, you feel that God knows where my brain has gone. I used to be so brainy, so sharp, and so intelligent once upon a time, and now in MBBS, I'm feeling that my brain has just degenerated. That is exactly what I'm trying to tell you, and this has happened to all of us, by the way. Even till date, I feel that how bright I was as a 10-year-old, it has just gone down from there, because the more you sit at one place and study, the more dull you keep on becoming. So you don't have to become like that. So when I say L form, it means either the bacteria had a cell wall, dhire dhire the cell wall went away. Option 1. Second, the bacteria does not have a cell wall ever in his life. So the L forms are of 2 types, unstable L form and stable L forms. Stable L form basically means that is seen with a bacteria called Mycoplasma. This means that there is no cell wall. It is going to be that, I will first tell you that you will never be able to see it. With your eyes, can you see bacteria? You can't. Can you see that do they have a cell wall or not? You can't. So all these tests are going to be done in the microbiology lab. When I was doing some tests, I found out there are some unstable L forms and there are some stable L forms. Let me actually start from unstable. What I do, I take a bacteria and I make it react with an antibiotic called penicillin. जैसे ही I made it react with penicillin, the cell wall will go. Have you read, in pharma, have you reached reading about penicillin, or are you still in general pharmacology? When you will study penicillin, they will tell you penicillin breaks the cell wall. It's a beautiful drug which can break the cell wall. So as soon as I put a bacteria with penicillin, the cell wall will be gone. But in a unstable, क्यों बोला? Because once the action of penicillin will go, drug का concentration खत्म हो जाएगा, they can revert back to their original form. They will get their cell wall back. So till the time, it's like a principle. Penicillin is like a principle. Till the time the principle is there, all the students will be scared. They will lose their cell wall and they will become innocent. As soon as the principle will go, penicillin will go, all of them will get their cell wall back. So they are what? They are unstable. They are not keeping in one form. They are going no cell wall, yes cell wall, no cell wall, yes cell wall. They are dodging left, right. That is why they are unstable. Unstable L forms are of two types, protoplast and spheroplast. Protoplasts are usually seen with gram-positive organism in which the entire cell wall is lost. As soon as I added penicillin, full cell wall gaya. So for example, if this is a bacteria and this bacteria had a cell wall, as soon as I added penicillin, that entire cell wall, पूरा का पूरा cell wall ended up vanishing. That is going to be protoplast. How I have learnt it is P for protoplast, P for it is seen in gram positive, and पूरा cell wall is going to be lost. Entire cell wall is lost. What is spheroplast? Ulta, it is seen in gram negative, and in this only some part of cell wall is lost. Means if this was the cell wall originally, now what will happen? Only some part of these cell walls are going to get lost in between. Then it will become a spheroplast. So repeating, what are unstable L forms? That ma'am, when you add penicillin, they will lose their cell wall. When you remove penicillin, they will get their cell wall back. That is why they are unstable. Protoplast L form is seen in gram-positive bacteria where entire cell wall is lost. Spheroplast is seen in gram-negative where some part of cell wall is lost. These were unstable because they were going to and fro. What are stable L forms? That once they lose their cell wall, they will never come back, unable to revert to original form, and that is mycoplasma. Mycoplasma, by the way, is one bacteria which is बिल्कुल अलग. सारे बैक्टीरिया एक तरफ, माइकोप्लाज्मा एक तरफ. Do you know mycoplasma? SMA is the smallest bacteria that we know. Number one, a very important quiz trivia point about it. It's the smallest bacteria. Second, it is a stable L form. Once it loses the cell wall, it is never going to get its cell wall back. So you have also learned what are L forms. I will give you a recap so that you remember it for your exam. Please note that when you're talking about cell walls, cell wall chala gaya, that is known as L forms. I'm giving you a mind map now. L forms are of two types. Number one, ma'am, they are going to be unstable. Unstable means to and fro. Chale jayenge, aa jayenge, chale jayenge, aa jayenge. These are of two types. One is known as protoplast and one is known as spheroplast. The next one that you have is going to be the stable L forms. Once they go, they will never, the cell wall will never come back. And the only example that you have to know under this is going to be mycoplasma. If you make this kind of a flowchart in your exam rather than writing it like this, examiner will get a better outcome. So make one summary table. Whenever you explain something confusing, make one summary table. Now I want to go back because major lengthy part of it is done. The first thing that I had on the outside was what? Capsule. Pad liya ma'am. After capsule, ma'am, you said the next thing you would teach us, lengthy one was going to be cell wall. Again, pad liya. Now let's go further. I can notice the third layer that I have over here is going to be cell membrane. And then let's trace it. Let me draw a cell membrane. This is a cell membrane like this, and I can see cell membrane is invaginating. Can you all notice? Cell membrane is going inside. What is this known as? This is referred to as the mesosomes or chondroid. Again repeating, it is invagination of what? It is invagination of the cell membrane, that is known as mesosome or chondroid. You will ask me, kya hota hai? It is a respiratory unit. Like, what do you use for breathing? You use lungs. So for human beings, there are lungs, right? But for bacteria, what is there for breathing, for respiration? They are the mesosomes or chondroid. Are they seen in gram positive and gram negative? Yes, they are seen in gram positive and gram negative. Breathing to sabhi ko karna hai, but they are more commonly seen in gram positive. So tell me the three questions of mesosomes. Question 1, what is the function of mesosome? It is a respiratory unit. Is it seen in both gram positive, gram negative? Yes, but more in gram positive. And it is formed because of the invagination of cell membrane. Are we okay with mesosome? All three questions. Now let me go. I'll keep going back to that diagram again and again. Cell membrane bhi ho gaya, mesosome bhi ho gaya. Now I notice, ma'am, there is one nucleus over here. I'll first label it. I can notice there is one ऐसा circular सा ऐसा nucleus over here. Then I can notice there are some dot 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 ribosomes over here, and then I can notice there are some miscellaneous inclusions over here. These are the three things more that I can see. So let us start reading. When I say that there is a nucleus over here, yes, what is the shape of the nucleus? I'm always making it circular. Am I making one circle or am I making two circles? I'm always making two circles. देखो हर बार दो दो color का circle आ रहा है, because which DNA is it? It is a double-stranded DNA. Have you understood the nucleus of a bacteria? Double-stranded DNA. Okay, apart from that, I could see a lot of ribosomes. Which kind of ribosomes does the bacteria have? 70S ribosomes. And what is the third thing that I could notice? Inclusions. Does every bacteria have inclusions? No. Just as an example, there is an inclusion called volutin granules. Right now you might find this name very new. Volutin granules are going to be present in a bacteria called Corynebacterium diphtheria. क्यों दिया मैम? ये क्या है inclusions? Volutin granules are nothing but energy stores. Like what is the energy in you guys, in us? ATP, मैम. ATP powerhouse mitochondria होता है हमारा energy. It has all the ATP production ability. Same way for bacteria also, they also need energy. So whereas Corynebacterium diphtheria stored its energy, it's in volutin granules. So these will be the inclusions, and you are settled coming back all the way and making you revise कि मैम ये था nucleus. अब बताओ nucleus कैसा होता है? Nucleus is double stranded DNA. राइबोसोम्स कैसे हैं? 70S राइबोसोम्स. इंक्लूजन्स में कौन सा? वॉल्यूटिन ग्रैन्यूल्स. खत्म. व्हाट आर द टू थिंग्स लेफ्ट? आई कैन सी वन बिग थ्रेड कमिंग आउट, दैट इज नोन एज द flagella, and I can see some tiny threads coming out which are known as fimbriae. So the last two things left are going to be flagella as well as fimbriae. Ready for their discussion? Because after that we would almost be done. Flagella ही बचा है, which is fimbriae is 2 lines. Flagella is the main. So first let me draw one flagella. Usually when they talk about the length, it is 3 to 20 microns in length. And look at the thickness. That's why I said it's like a hair, because even if you don't learn the thickness, it's 0.01 microns. It's very, very thin if you overall look at it. Exactly like a hair, and the main function of it is going to be locomotion. Movement will be available with it. Plus, it is also going to have an antigen called H antigen. Now I want to ask you a question. Have you all heard of a disease called typhoid? I mean, that's a very stupid question. All of you have heard of a disease called typhoid. Do you know what is the test which is done for typhoid? Whenever someone suffers from typhoid infection, कौन सा blood test करते हैं? We do Vidal test. Vidal test tells us about antibodies present in the blood. If anyone, typhoid is an infection where everyone has food poisoning. कोई बाहर का street food, bad water, golgappa, pani puri you had, it had dirty water in it, contaminated water. You ended up eating and drinking that water, and after that you have landed up, not you, but someone has landed up with some typhoid, food poisoning. What test would you want to do? You will say that, okay, I'll go give my blood test, and in my blood test they will find antibodies. That test is known as Vidal test. Antibodies against which antigen are seen in Vidal test? O antigen and H antigen. Now you will tell me, where was O antigen present? कहाँ पढ़ के आए थे मैम? ओपा पढ़ा था. ग्राम नेगेटिव के लाइपोपॉलीसैकेराइड के सेल वॉल में पढ़ा था O antigen. And where are you studying this H antigen? The H antigen is going to be present in the flagella. So now tell me, who causes typhoid? Typhoid is caused by Salmonella typhi. That is how you, why do you guys find bacteria so tough? Because you don't study the general. If I ask you, Salmonella typhi, is it a gram positive organism or gram negative? You will say, ma'am, if Salmonella typhi has O antigen, OPA, matlab it is gram negative. Okay. Does Salmonella typhi have a flagella? Of course, because if it has H antigen will come from flagella. Salmonella typhi is a gram negative organism. It has flagella. The test is known as Vidal test. Half of Salmonella typhi you have done. So coming back, flagella is 3 to 20 microns in length, very, very thin. It is going to help in movement. It is also going to have an antigen. This is how a flagella looks like, whether gram positive or gram negative. This is how it will look like. You will have one thread coming out. That part is known as the filament. I'll write it again because the font is small. Then you will see it takes a turn like this. That is known as the hook. And then it goes into the cell wall, and that is known as the basal body. That is known as basal body. So guys, whenever you have to draw a flagella, you will make a cell wall like this. In that cell wall, you will say the part in the cell wall is going to be basal body, and you can see the basal body is having some rings. Okay, the basal body is going to have some rings. Then one turn comes out, that is the hook, and then finally that free end comes out, that is filament. What are the three parts of flagella? Basal body, hook and filament. But ma'am, how will I know this is gram positive ka flagella ya gram negative ka flagella? So you will have to look at the number of rings that are being made. In a gram positive flagella, there are only 2 rings that are made. That means, what are the names of the rings? M and S ring. M ring, S ring. So Miss positive is what you will learn. Miss positive. So Miss positive means 2 rings hai. M and S is gram positive bacteria. If you have 4 rings, MS तो होगा, साथ में if you can learn the alphabets are not important. M, S, P, L. If there are 4 rings, then that is going to be gram negative. So let me come back. Over here, I had drawn 4 rings. So can you now tell me what is this? Ma'am, if there are 4 rings, let's assume M, S, P, L. 4 rings are going to be gram negative. If there would have been 2 rings, that is MS, then it would have been gram positive. Okay, this flagella, look at the parts. This has rings, this must be the basal body. Then I have a turn, that must be the hook. And then I have the free element, that must be the filament. Basal body, hook, filament. Basal body, hook, filament. Yaad ho gaya? 3 parts are done. Gram positive, gram negative is done. Flagella is done. What do you mean by endoflagella? Flagella which is inside. Where inside? Ma'am, you just now told me it was in the periplasmic space, and it is seen in which organism? It is seen in spirochetes. ये तो हम already लिख के आए हैं, that Treponema pallidum. So this we know. Now we will understand what is the location of the flagella. यहाँ पर आता है mugging up part. I just told you, oh, it's a thread coming out. But from where? Listen carefully. This is a bacteria. How many flagella do you see coming out? Only 1 flagella, ma'am. That is why I call it mono for 1, and trichous. Tricho का क्या मतलब होता है? Tricho means hair. So flagella looks like a hair na. 1 hair coming out, 1 flagella coming out, monotrichous. Okay, just get used to the names. In the next bacteria, I see 1 flagella coming out on the left and 1 flagella coming out on the right. So both ends. This is known as amphitrichous. Acha, what does that mean? Trichous means hair. Amphi means what? So usually, I don't know how many of you remember this, but we used to study that there are some people who can write with their left hand also and with their right hand also. They are known as ambidextrous people. Ambi, amphi, ये सारी चीजें तब use की जाती हैं when you are dealing with left and right both. So amphitrichous means 1 flagella on the left. Anyone in class today who's ambidextrous or was ambidextrous for at one point of time? Because then you would have some similarity with me. I remember that for a very long time, I think 8 or 10 years of my life, I was ambidextrous before I became left hand dominant. And after that, I have only used my left hand, but I wish, like Virus in 3 Idiots, I would have also continued writing with both my hands. But there are a lot of people who carry that habit forward and can be ambidextrous for the rest of their lives also. It's just about how you train your brain, right? So ambi always means 2 sides. Over here you're using amphitrichous, so it's 2 sides. Okay, if I say there are 4-5 flagella, like aisa hair tie kar diya hai, you made a ponytail out of it, that is known as lophotrichous. On one end, in simple words, you can study lophotrichous is going to be a bunch. Oh, so I have someone with me over here. Dr. Tushar, if you are ambidextrous, but then what's your dominant hand? Are you a lefty or are you dominantly a right-handed individual? But in case you're lefty, then high five again. Okay, anyway, that I think would be another class on another day where we could have non-academic conversations, which I am assuming are going to be less sleepy and less boring than microbiology. Okay, so lophotrichous means 1 bunch on one side. Now what are we left with? Peritrichous. Peritrichous is going to be periphery. The flagella are present all around the periphery. Now I feel the words are so simple. Monotrichous means 1, amphi means ambidextrous left and right, lopho means it's going to be a bunch, and peri means it's going to be on the periphery. Meanings are done, but now I have to study what is going to be their underlying example. So for monotrichous, your example are Vibrio, cholera and Pseudomonas. कैसे याद करें? Vibrio sounds like 1. Vibrio sounds like 1. And Pseudomonas also sounds like pseudomonoas. So now you will remember Vibrio has how many flagella? One flagella, monoflagella. Pseudomonas has how many flagella? One flagella, monoflagella. याद रहेगा Vibrio 1, Pseudomonas 1. Amphitrachus you don't have to learn any, so good for us. Lophotrichus is what? Bunch. Ma'am, Bacters have a bunch. You must have heard of Helicobacter pylori. With which organ does it go into? This bacteria, Helicobacter pylori, always goes into the stomach. Helicobacter pylori, the bacter, remember bacters are going to have a bunch, Lophotrichus. Jo bacha, that is going to be Peritrichus. So Peritrichus tells me the name itself, Peritrichus, is going to be PR for Proteus and E for E. coli. Listeria you don't have to learn. So somewhere you will have to put a stop in micro, otherwise it's an endless list of everything. I'll give you a quick recap without us looking at it. Monotrichus ke example batao. Mono means one, ma'am. It is Vibrio 1 and Pseudomonas 1. Then if it is present on both sides, it is known as Amphitrichus. If it is present as a bunch, that is known as Lophotrichus. Bacters have a bunch, Helicobacter. And if it present all over the periphery, it is known as Peritrichus, which is seen in Proteus and it is seen in E. coli. Location yaad ho gaya? I hope you're not just reading, you're learning with me in class and going back. So location is also done. Now you will ask me, how do the bacteria move? What is the types of motility that you have to know? Very simple again, because right now I've got you a list of the entire bacteria, parasites, sare locomotions I have got for you over here, which will make it a lot easier for you going forward. So let us get going to the types of motility. Tumbling. Tumbling kya hota hai? Tumbling means like a drunk man. He falls on the left. Someone who's very, very drunk falls on the left, falls on the right, cannot walk in a straight line. Drunk man, who's that? Any friend of yours who drinks a lot and who shouldn't, next time tell him or her that he or she is behaving like Listeria. Listeria shows tumbling motility, falls to the left, falls to the right. Listeria also shows differential motility, which means Listeria has a temperature that if you put me at 22 to 25 degrees, then I will show you tumbling motility. Then I will show you falling left, right. But if you put me at 37 degree, it's too hot. I don't want to drink. I don't want to tumble. So there is going to be no motility. So can I say this is a difference created that at one temperature it will show motility, at one temperature it will not. I will give you one life lesson. Ideally, you should not drink at all. It's very harmful. But if you have a friend who is drinking, always give him one lesson that listen, right now you are in your 20s. In your 20s, your liver will support you. You can fall here and there and it's okay. But once you go in your 30s, late mid 30s, then you will not be able to drink so much and your liver will not support you. You will not be able to undergo tumbling. So at a young age, you can afford to tumble, but at an older age, you cannot afford to tumble. Okay, so Listeria shows you tumbling and differential motility. Okay, next one is who shows you darting motility. Darting motility is something known as shooting star. You must have seen, especially in Bollywood movies, Tootta Tara, shooting star. You can't see it only. It is so quick. Within half millisecond, it runs away. That is Vibrio and Campylobacter. You can't see them. They are so quick under the microscope. I cannot see them. I just feel someone going whoosh, whoosh from here. That is how fast they go. Next, corkscrew motility. Corkscrew means you all have seen how a screw is. It is going to move round and round and round. Correct? Okay. Which is that bacteria that looks also like this? Like it was looking also spiral. Ma'am, you had just taught us spirochetes. It looks also like Treponema pallidum is a spirochete. It looks also spiral. It moves also spiral. See, it looks also spiral and it is going to move also like a corkscrew motility. Coming to twitching. Twitching means, what is twitching? All of you, sometimes have you ever experienced your eyelid starts, wo aankh fadakna, what you call it? Your eyelid starts twitching or some muscle of yours starts contracting, twitching. That kind of 1 second movements. Those twitching motilities are shown by trichomonas, T for T. Okay, then which of them shows you falling leaf motility? Falling leaf like the Shah Rukh Khan movies, like Mohabbatein and other Shah Rukh Khan movies, where there is a setup of Canada, there's a lot of snow, and then those leaves are falling aram se in the air, they come down. That kind of chilled out falling leaf wala motility, that is going to be shown by Giardia lamblia. How I have learnt is Giardia lamblia is girta hua leaf. Giardia lamblia is girta hua leaf, so falling leaf motility. Now we are just left with swarming motility. Swarming means the picture that is shown over here. This is of swarming motility. You can see that there is concentric circles. That is how the bacteria is moving and growing. The bacteria is moving in concentric circles. I'll erase it so that you can see it yourself. This is swarming motility, and swarming is shown by anyone. Swarming is shown by which bacteria? It is shown by PVCs, multiple bacteria, but that's the last lengthy list that you have to learn. What is the first one? It is going to be Proteus. The next one is going to be Vibrio, वापस जाओ P से, Parahemolyticus. Vibrio Parahemolyticus. The next one is going to be Clostridium tetani, and the last one is going to be Serratia. Repeating, who are the 4 bacteria that can show you this round and round and round swarming? It is going to be Proteus, it is going to be Vibrio Parahemolyticus, it is Clostridium tetani, and it is going to be Serratia. I just want you guys to repeat this without looking at it. If I ask you, so best is that I erase this all together. If I ask you, for example, over here, okay, so it's the erasing is not working. Anyway, if I ask you, tumbling motility is where? Tumbling motility is Listeria, drunk man. He will tumble at young age 22 to 25 degrees. He will not tumble at 37 degree. Who will show you darting motility like a shooting star? It is Vibrio and Campylobacter. Who will show you corkscrew motility? Looks like a screw, moves like a screw, Treponema pallidum or spirochetes. T for T, twitching motility, Trichomonas. Swarming ka only tell me the mnemonic, PVCs. Swarming motility, round and round wala, is known by PVCs. Falling leaf, girta hua leaf, Giardia, Lamblia. All the motilities are done. Now your next question is, how am I going to demonstrate the motility? There are, you are saying, ma'am, this is moving like a star, this is moving like circles, like a screw, but how do you see it? In a lab, how do you see all of that? There are multiple methods. The most commonly that we are using nowadays is known as the hanging drop motility. So first let me explain hanging drop motility to you. What do you do? And you will be doing, have you guys done hanging drop motility in your practical lab as of now? If not, you will be doing this further. So what are you going to do? In this, you will actually take this over here is a cover slip, right? And नाम से क्या समझ में आ रहा है मैम? नाम से I can see that there is hanging drop. I have to create a drop that will be hanging. Exactly वही करते हो. What you do first, this is a special slide that you will get in your lab, and this slide will have a central depression. मतलब ऐसा वो usual glass slide नहीं होगा. इसके center में से गड्ढा होगा. There will be a depression. What will you do? This slide you will keep on one side. Then you will take a small glass cover slip. On that cover slip, you will put whatever bacteria you want to study. You want to find out, is Listeria motile? Does it show tumbling motility? Is this Listeria or is it not? So you put that organism over here, and ulta karke, now you have put that cover slip onto that slide. So are you making this drop hang from above? Are you talking about hanging drop motility? So this is the slide, and on this cover slip you have put the bacteria, and now you are making it hang ulta karke. That is what you call as hanging drop motility, and you will be able to see whether it is moving like a star or whether it is going in a screw manner or whether it is falling leaf. So that is the best way to identify that you will do hanging drop motility. Of course, abhi I am not going into how to do practical things, wo ek alag class hoga when the right time comes, but this is just to tell you that these are the tests. Second, you can grow it in agar. Agar क्या होता है? For those who didn't notice, did you notice what was this? जहाँ swarming दिखाया था, this was actually blood agar. This was blood. It's solid. Agar is solid. Yeast से आता है. So now, if I take, let's say, agar 2%, you'll say, ma'am, 2% concentration is too much. If someone wants to move around, and if you keep a very tough environment, how will they move around? थोड़ा सा concentration कम करो ना, then everyone will move around. Use a semi-solid agar. Semi-solid agar has a concentration of only 5% agar, so the bacteria will easily be seen moving around. The next one, there are 2 tubes you can use. From there you can actually see the bacteria will be seen on the surface moving. Either you have a big tube and then a small tube inside it, that is known as the Craigies tube, or you actually have a tube which looks like U-tube. In both of them, you can see the bacteria moving. So what are the 4 methods of seeing the motility? Either you can invert the drop, that is known as hanging drop motility, और you can use semi-solid agar. Semi-solid क्यों use करा? Because 2% agar तो बहुत strong, rigid था. Bacteria could not move. It said environment is very tough. Make the environment a little soft. So you used semi-solid agar of 0.5%. Thereafter, this tube within tube is known as Craigy's tube, and this is known as the U-tube. 4 methods of demonstrating motility. Now, for that extra edge question, that INI-CET level or distinction level question, what is the special stain that you have for flagella? And that is the Liefsen and Rieu's stain. I have not taught you under stains because koi puchega nahi. It's like if you only want that short note to have that extra edge, Liefsen and Rieu's stain is for staining flagella. Again, how I have learnt it, Liefsen will remind you of locomotion, right? And Liefsen is for flagella. So locomotion कौन कराता है? Flagella कराता है. तो Liefsen and Rieu's stain is going to be for flagella. Okay with everyone? Moving forward, flagella done. Now we are left with the smaller ones, that is Fimbriae. I don't know if you remembered, but the last thing after flagella, the small wale 3 structures, what are those? Those are going to be the Fimbriae. And here you get going. What are Fimbriae, ma'am? The last thing that you have to know for today. Fimbriae are very, very tiny, like a hair. If I draw it again, they are coming out like hair. So do they have an antigen? Yes, they do. Now I want to ask you, did flagella have an antigen? Yes, what was the name of the antigen that flagella had? It was H antigen. Do Fimbriae also have an antigen? Yes, they do. But there is no name given to their antigen. Bas antigen hai. What is their main function then, ma'am? For flagella, the main function was locomotion, movement. For Fimbriae, the main function is adhesion, sticking. Whenever bacteria comes in my body, how can it stick? It can stick by Fimbriae. If one bacteria wants to stick to another bacteria, both of them want to stick to each other. Let's say, जैसे humans में male, female, there is in animals there is mating, in male, female there is reproductive cycle, so there has to be mating and meeting of the male and female. Na bacteria में भी same होता है. There is a male bacteria, there is a female bacteria. If they have to combine or if they have to mate, what you don't use the word mating for bacteria, but if they have to undergo reproduction, they will have to connect. How will they connect? Is by Fimbria. So if this is one bacteria, for example, let's say this is a male bacteria, and this has to adhere to a female bacteria. In between them, that connection will be made by Fimbriae. And because it's between male and female, instead of calling it Fimbriae or Pili, can I now call it sex pili? Because this is causing an attachment between a male and a female. So now I call it sex pili, and this process of sex pili formation in bacteria is known as conjugation. Repeating before I tell you the last part, what are they? They are hair-like. Yes, ma'am, they have an antigen. Their main function is adhesion, chipakna, to stick. And when the sticking happens between a male bacteria and a female bacteria, instead of calling it just a pili or a Fimbriae, I try to call it a sex pili. Now my next question is, other than adhesion, can it sometimes help in something else? Please note, can help in locomotion, can help in locomotion. And you will be so shocked. You will say, ma'am, right now you told me locomotion के लिए तो वो बड़ा वाला flagella था. Now you are saying Fimbriae can also help in locomotion. That is a very weird thing, but where does it help in locomotion? There is a special type of Fimbriae called type 4 pili. They are going to help in locomotion of which two bacteria? One is going to be Neisseria and the other one is going to be Pseudomonas. In these two, this was, by the way, this was the recent 1 year ago INI-CET exam question, and no one will ask you in Viva especially, but when you write a short note on Fimbria, you should be writing this. That Fimbria otherwise are for attachment, adhesion, sex and all of that. But is there any bacteria where Fimbria, these tiny threads will help in movement for two bacteria, Neisseria and Pseudomonas. So what are those Fimbriae known as? They are known as sex pili. What are the tests that we do for Fimbria? How will I get to know that there is a Fimbria that are present? Again, you don't have to know the details, just the names. One is known as the hemagglutination test. One is known as the surface pellicle test. Trust me, no longer these are done, and you do not have to go into the details of any of these. Hemagglutination test and surface pellicle test. That's it, nothing more required. I'll just do a repeat of tests of flagella and fimbriae and then we are done for the day. Flagella के tests कौन कौन से थे मैम? You told us एक तो hanging drop motility था, semi solid agar था, Craigy's tube था, U tube था. This was for flagella. For fimbriae we have hemagglutination test and surface pellicle test. And finally with that we have finished the entire. Let me just ask you very quick questions. If I say what was outermost, ma'am, outermost was capsule or slime layer, which is that one bacteria which can have both. Streptococcus salivarius can have both capsule and slime layer. Thereafter you had cell wall. For cell wall you had to remember PPPT because gram positive cell wall has more of peptidoglycan. It is going to have pentapeptide bridge. It is going to have teichoic acid. Then you came to cell membrane. वो जो invaginate करा, these were mesosomes, the respiratory one. Then you had a nucleus which was double stranded, you had ribosomes which are 70S, there were inclusions like volutin granules. Then you had a long flagella, that flagella was for locomotion and it was going to have the H antigen. On the other hand, you had fimbriae. Fimbriae were just for attachment. अगर male female में attachment हुआ तो you call it sex pili, and fimbriae can also have an antigen. That's your basic summary that you had to know, with which abhi you have finished microscopes today, you have finished bacterial anatomy today. Next class, of course, we will be taking up all of this. So you have 2 more classes for general micro left, which is going to take care of whatever I have segregated accordingly. And yeah, we'll be meeting now in the new year in 2025, where I've already told you there are going to be 3 classes of path and there are going to be 3 classes of micro. Of course, general micro we will finish off and we will start with bacteriology or the systems also. In pathology, we will finish cell injury and we will finish most of inflammation. That is your plan in case you are planning to study. Cell injury khatam ho jayega, inflammation 80% khatam ho jayega, and microbiology mein full general will be done. Jan will be settled for us. I hope everyone is okay with this. No confusion, no problem. Before you leave, if you could answer 1 or 2 questions for me, that would actually be great. That's it. While examining a bacterial slide under the microscope, you notice that the organism lacks a cell wall. The cell wall deficient organism that could be seen on the slide is lacks a cell wall. Is it that L form? Cell wall deficient form. Which was the cell wall deficient form? Very good. Mycoplasma. One liner mein to achhe. Very good. Now little last question, lengthy question. There is a sputum sample which was sent for testing in the microbiology lab. The gram staining showed gram positive cocci which correlate with bacterial cell wall property. Which of the following statement is false regarding bacterial cell wall? मतलब gram positive cell wall के बारे में you have to tell me the false statement. Start reading with me. The cell wall of gram positive is thicker. Is peptidoglycan more? Is it thicker? Ma'am, I think this is true. Is teichoic acid present in gram negative bacteria? No. Gram positive, ma'am, you told us PPPT. Teichoic acid was present in gram positive. I have found the false statement. That is the answer that they asked me to search. Let me read other options. Region 2 of lipopolysaccharide is polysaccharide. I had learnt OPA. Region 1 was O antigen. Region 2 was polysaccharide. ठीक है. And region 3 was lipid. This is also true. Next, lipopolysaccharide is responsible for O antigen specificity. हाँ, O antigen variable होता है, तो O antigen is present again in OPA of lipopolysaccharide. O antigen is going to be present, so this is also correct. Yeah, don't worry. There are 2 more things which are still there, which I have not. Abhi, your story is not over. Someone asked me. So next class, when I'll teach you the physiology, in that I will be teaching you 2 more things which in your books is written under anatomy, but right now you will not understand anything. One of it is toxin, endotoxin, exotoxin, like you're telling me, and second is about spore formation. So both of these I will be able to tell you when I teach you the entire growth curve over there, where toxin occurs, where endotoxin production occurs, where exotoxin occurs. Also, like you're telling me, the differences we will study, and I will also teach you spore formation. So with the growth curve you will understand with the understanding. Otherwise it becomes very you're just mugging it up. Okay, so don't worry, that is going to come. Perfect. So I think we are settled with today's class. I have told you what all we'll be covering in the next class also, so you can prepare. For pathology, we'll be finishing off the rest of cell injury. Okay, so thank you so much guys. Okay, so I was not aware. I was, of course, you would be needing the 2x 3x in the later run, because every time you can't look at that entire video. So for Prof Edge, I wasn't aware that it's not come up into the team, into the videos. Dr. Vishal, give me some time. I will convey this to the team and let's see what can be done, because I totally get your point here. Okay, Dr. Pranav, you're able to solve in second year or third year, whichever prof you are in right now. If you are able to solve 70% of Q bank right now, it is good, because see, Q banks are not prepared keeping in mind second prof students. Q banks are always prepared keeping in mind competitive exams. So your path Q bank will always have an element of medicine, surgery, gynae. Your micro Q bank will always have an element of pharma, medicine, surgery. So when you have not studied those subjects, you will have a little bit of a problem in answering questions which might come from there. So like for example, I might teach you melanin in pathology, but they might give you an integrated question from dermatology asking you about melanoma skin cancer and then correlating it with melanin. So that is where you might not be able to answer the question when you are in your second or third prof. But don't take that like in your second prof you have zero clinical exposure right now. So don't take that you know you're, again, this is so no time for you to run after the accuracy of marks that you're getting in a Q bank. This is the time in you have to go into knowledge abundance, knowledge overload. Right now you neither have to make things concise, nor you have to think of that how can I make it into 20 pages, or how can I get a 95% Q bank score. You are in the very primitive starting stages. Now just take it as a learning curve. Even if you're getting 70%, that is not my concern. If you've picked up a topic and say it has 22 25 questions, you are doing all of them with their explanation, adding the extra points to your notes, learning something from the clinical aspect. If you're not understanding, see a relevant YouTube video, take advantage of social media in the correct format. If you're going and following that path, I think that would be more advantageous than just focusing on what percentage you are getting in the Q bank. This is not the right time. You come to me in internship with that kind of a score, I would say no, from 70 we have to jump to 85% plus. But right now I would say that is very good, because you are not got any clinical exposure. Even without that you are scoring 70%, that is great. So don't consider that as any parameter for the time being. You have your entire curriculum to run after marks. This is not the time, at least not for Q bank. It's totally learning mode that you have to get into. Okay, hematology 80% average. You are running opposite, by the way, because hemat is one thing which every student says that ma'am we are not even scoring 40%, and patho we can still manage 80%. So I think everyone has that way of sometimes learning, or there are certain topics you end up liking more. So you have ended up liking the beast or the giant, that is hemat. And what I would say, you would have to work a little bit more. And I hope if you've seen the main videos, if you're in that phase that you are way into your second year, you've completed a few months, then you can go to the main videos. If you have time, then you keep following the live batch, and then you keep doing the Q bank once again alongside. That will give you a little bit of confidence and will keep clearing your concepts. Okay, yeah, Dr. Shreya, and to everyone else, because we won't be meeting here after, firstly, Merry Christmas. It's a very bad day that I chose for a class, Christmas Eve, but I wanted to give you all off tomorrow, and that is why I kept the class today, that at least on 25th, I'm assuming all of you must be having a holiday tomorrow. So yeah, I didn't want to keep a class, and I'm not troubling you at all till New Year, and I'll be meeting you in 2025. So wishing you all a very, very happy New Year, well in advance to you and your family. Do make that New Year resolution that you will study every day, because your life is incomplete as a medico if you don't do that. It will be ditching your community if you don't make that resolution of being regular with your studies. So do make it, and I know you will break it because all of us have, but with a pinch of salt, we should still sit down and make that resolution for all of us, right? So wishing you all the very best. Thank you so much.